Okay. اوكي دكتور، حاليا نسجل. عندنا 31 اتنديز.
انطي 5 دقائق.
اي
دكتور.
نعم.
وعليكم السلام ورحمة الله وبركاته. من دكتور بلال؟
This meeting is being recorded.
Shall I start, Ustad Ara?
Go ahead, please, doctor.
Yeah. بسم الله الرحمن الرحيم. good morning us from the United States of America. For the rest of you, I know the timing is completely different. So good evening, everybody. Welcome all of you to the Modern Trends and Technologies in Pharmacy Research, our third virtual conference. My name is Dr. Inam Sameh Arif, Dean Associate for Scientific Affairs in the College of Pharmacy, Mustansiriyah University. On behalf of the staff and colleagues of the College of Pharmacy, Mustansiriyah University, we have the pleasure to welcome all of you. We are sorry that we are having this conference virtually, but as you know, it is because of the pandemic. During a couple of days, we are gonna focus on the recent advances in the field of pharmacy research and the use of technology in this field. Then we are gonna display the scientific efforts of the junior researchers, junior Iraqi researchers. So I would like to thank the speakers who are joining us from abroad, Dr. Peter Crooks, Dr. Zainab Al-Bayati, Dr. Brandon Frett from Arkansas University, and Dr. Raad Kash from Philadelphia University, and Dr. Lina from Jordan. And the same thank for our colleague, Dr. Ghida. I would like to thank also a big thank to the people who helped in organizing, preparing this event and make it possible. A special thank for Dr. Ghazi Al-Tamimi and Dr. Zainab Al-Bayati for their help and support for preparing and organizing this conference. I hope that during these couple of days, we are gonna have a nice, useful, and interesting time, discussions, exchanging ideas about pharmacy research. Finally, it is my pleasure to introduce Dr. Munder Faisal Mehdi, Dean of the College of Pharmacy, Mustansiriyah University, to give her talk. Dr. Munder, the stage is yours.
Assalamu alaikum wa rahmatullahi wa barakatuh.
عليكم السلام.
Good afternoon for everyone. Good morning to others. Bismillah ar-Rahman ar-Rahim. It is my great pleasure to welcome you to our third International Annual Conference. Firstly, allow me to express my gratitude to the President of the University of Mustansiriyah for his tremendous support. Secondly, we were very grateful to the conference organizing committee whose members have put together an astounding event. On this platform, I would like to take the opportunity to reintroduce the College of Pharmacy, despite its young age, which was established in the year 2000. It has proven its presence through the rapid development of the college's infrastructure and the substructure of the classrooms, green spaces, laboratories, and equipment. Regarding the college's scientific staff, the college has and continues to support its teachers to make their ambitions happen and complete their higher academic degrees, nationally and internationally. Many of our teachers have obtained their master's and PhD degrees from the US, UK, and Malaysian universities. As the college's infrastructure, in 2014, a large new building consisting of 12 scientific laboratories computer science laboratory, math library, a special student club, as well as staff and teacher rooms have been added to our college. In 2011, we were able to offer our first master's degree in pharmacy sciences. Since then, we continue to open up more courses that now we offer degrees in pharmaceutical chemistry, laboratory sciences, and pharmacology and toxicology, as well as the PhD in pharmacy. Starting from this year, we will be offering higher studies program in clinical pharmacy and pharmaceutics. Lastly, I am very proud to speak of the college's contribution as the pioneers in introducing the Cambridge Structural Database, CSD, software package to our beloved country, Iraq. CSD is the world's repository for small molecule, organic, and metal organic crystal structures, containing over 1 million structures from X-ray and neutron diffraction analysis. This unique database of accurate three-dimensional structures has become an essential resource to scientists around the world. A research team of me and two of my master's students, Noor and Omar, was the first to purchase the legal rights and obtain the license of CSD Gold, which means genetic optimization for ligand docking software from the University of Cambridge in 2016. Ever since, the license is being annually renewed, and more than 10 of the workshops have been organized all over the country, inside and outside the University of Mustansiriyah, to introduce the software to researchers so that they can learn how to properly utilize it and make the utmost benefit in their work. This program enables higher studies students working in research field in examining the features and behaviors and the impact of molecules on various compounds, which helps in the prediction of different molecules binding modes in a three-dimensional structure, which is a problem of paramount importance in rational drug design. In brief, GOLD is an automated ligand docking program that uses a genetic algorithm to explore the full range of ligand conformational flexibility. Visualization, it is one of the top 3 software in the world, with accuracy near 95%. Our college has intensified its efforts in developing and validating this program in Iraq, and ever since has attracted researchers from all over the country to apply molecular docking in their research. Finally, in conclusion of my speech, I think all those who contributed in preparation for this conference, and I hope you are satisfied. Now I will leave you with the first lecture. Thank you for listening.
Thank you, Dr. Munder. Now we are happy to start our first scientific session, which is entitled Computer-Aided Drug Development, which will be coordinated by Dr. Wathiq Alwan and Dr. Samra Karagholli. Good luck, our colleagues.
Good luck.
Thank you very much, Dr. Alwan.
Thank you very much, Doctor.
Good afternoon, ladies and gentlemen. Good morning to our guests abroad. First of all, I would like to thank each one of you for joining us in our third international virtual conference titled Modern Trends and Technologies in Pharmacy Research, which is organized by College of Pharmacy, Mustansiriyah University. I bid a very warm welcome to all the speakers who took out their valuable time and joined us today to be a part of this conference. We are honored to have you all with us. My name is Dr. Wathiq Alwan Al-Hachami, and I will chair with assistance of my colleague, Dr. Samra Al-Karagholli, the first session, Computer-Aided Drug Development. Just giving you a quick overview about this session. This session consists of two parts. In the first part, there will be 3 talks, and then followed by a Q&A session. So feel free to drop your question in the chat box, or you can raise your hand after finishing 3 lectures. Then after a 15 minutes break, I will leave the platform to Dr. Athmar Zahar and Dr. Ola to manage the second part of oral presentation. So I think we have to start now. Let's get started with the first guest speaker for this session. Dr. Samra will introduce our guest speakers in this. Samra, the platform is yours now.
Thanks. Thank you very much, Dr. Wathiq, for this introduction, quick, sharp, and nice. My name is Samra, Dr. Samra, lecturer at Pharmacokinetics Department, College of Pharmacy, Mustansiriyah University. Our first speaker in this session is Dr. Ghaida Hamid. Dr. Ghaida, in 2001, she received her bachelor degree in pharmacy from College of Pharmacy College University of Baghdad. In 2009, she received her master degree in pharmacy, College of Pharmacy, Baghdad University too. And in 2017, she had her PhD from University of Nottingham. Currently, she is a lecturer at Pharmacy College since 2002. She has a quite area of interest. She is interested in drug formulation and pre-formulation, crystal engineering, and drug delivery. Dr. Ghaida will talk about in silico modeling in pre-formulating studies. Dr. Ghaida, she is my colleague. You have around 15 minutes to take us through the topic. Before starting, I would like to ask the audience to keep their phones silent, please. If you have any question, feel free to drop your question in the chat box or reach me or raise your hand, and you will have the time to raise your question. Dr. Ghaida, the mic is yours. Thank you very much.
Assalamu alaikum. Thank you, Dr. Samra, Dr. Wathiq. Thank you for Dr. Munzar and Dr. Am for organizing this conference. Sharing my presentation. Is it clear?
All is good.
Okay. Thank you very much. Ladies and gentlemen, good afternoon. My name is Ghaida. Today, I'm going to talk about in silico modeling in the pre-formulation studies. Today, I'm going to talk about the biopharmaceutical classification system, amorphous formation, hydrogen bond, the chemistry structure database, processing, and the prediction of hydrogen bond formation, and finally, give a conclusion. As we know, the drugs are classified according to the biopharmaceutical classification system into 4 categories: class 1, 2, 3, and 4. Class 2 and class 4 are the most important part because it's represent about 75% of the drugs. These are under researcher focus because of the low solubility, which is a big problem because it will give us a poor bioavailability and poor pharmacological activity. So the researcher focus on these 2 classes of the drugs in order to improve the low solubility issue. A researcher introduced many methods to increase the solubility of class 2 and class 4 drugs, such as amorphization, salt formation, lipid formation, co-crystal, pH modification, complexation, and surfactant. Amorphization is the most important part because it's increased the solubility of poorly water-soluble drug to about 1,000 times. What's amorphization? Amorphization is the conversion of the drug from the crystalline form into the amorphous form, which is the more soluble form. This can be done by 3 main methods, which is solvent evaporation, mixing the drug with and polymer with the solvent, then evaporate the solvent, hot melt extrusion, melting of the drug and polymer, then rapidly cooling them, and finally milling, which is an intentional process for transformation of the drug from the crystalline to amorphous form. Here is the sugar crystal, which is the simple example to explain how the drug is converted from the crystalline form into amorphous form. This is a lollipop. So the sugar crystal is melted, then it's rapidly cooling to form this interesting lollipop. So this is a simple example to explain this transformation. Usually, amorphous formation is by incorporation of the drug into the molecular dispersion, either by incorporating the drug into other drug molecules or into a polymer to produce an amorphous form. This amorphous form is highly energetic. It's the most soluble form than the crystalline form, but unfortunately, it's unstable form. Because of its high energy, it's going to be converted and returned back to the crystalline form. This recrystallization may be happened during the processing or after formulation. So this will be a big problem and big issue in the formulation. The most dangerous issue is the conversion from one polymorph to another, or making a different polymorph, which will cause a different solubility, and this will cause a low bioavailability and, of course, lower pharmacological activity. For this reason, there is a lot of theories to increase the stability of this amorphous form, such as increasing the glass transition temperature, adding a physical barrier, or through drug polymer interaction, etc. We will focus today about with a drug polymer interaction because it is the body of our talk, which is the chemistry structure database. So the drug can be formed like this drug, the first drug, sorafenib, polyvinyl pyrrolidone, solid dispersion, can be make a strong hydrogen bonding with polyvinyl pyrrolidone polymer. This will be more resistant as the drug polymer interaction is strong through hydrogen bonding. So this will be more resistant because the polymer will be surface enriched. Other drugs, B, mixing with the polyvinyl pyrrolidone, because of the presence of this F group, this will make a weak probability of hydrogen bond formation. So the drug will be on the surface of this dispersion and will be recrystallized easily. So now weak and strong hydrogen bond. How can we predict this hydrogen bond? It's important. So how can we predict it? They can predict it by the database, which is called the chemistry structure database. What's the chemistry structure database? It's the database that established in 1965. It includes small molecules, organic and metal organic crystal structure, million structure from X-ray and the neutron diffraction was analyzed. The molecules and the drugs was grown up since 1973 till now. Now we have more than 1 million compounds introduced into the chemistry structure database. How the materials and the drugs enter to the chemistry structure database? First of all, the industrial or pharmaceutical, from the pharmaceutical chemistry department, the new drug or a new pharmaceutical active ingredient is introduced into the chemistry structure database. Then it will be checked by the editors. After that, after checking, it will give chemical representation, bibliographic, and chemical physical property information will be given. Then academia, industry, new drugs will be get benefit from this entry. More than 50,000 new structure each year enter into this chemistry structure database with a regular update online. What's inside chemistry structure database? It contains a lot of organic and metal organic compound, polymeric and non-polymeric compound, single component, multi-component system, organic like drugs, agrochemicals, pigment, explosive proteins, metal organic framework, model for new crystals, additional data like melting point, polymorph, melting point, crystal color, shape, bioactivity details, natural source data, and oxidation state. And it will be linked to many sites such as drug bank, drug-like, MOF. All these sites for chemical compound are connected directly to the chemistry structure database. First of all, how can we introduce the motifs inside the chemistry structure database? First of all, we have our drug and polymer, which have either a donor and acceptor group. First of all, we should detect these donor and acceptor groups before entry. Then these two motifs, the acceptor and donor group, will be entered to this database through two main programs, which is Conquest, which is the database search, and Mercury. They are these two programs inside Cambridge structure database are work together. So we're building queries and combine these queries, then searching setup, then viewing our result. For more details, queries are building from the donor and acceptor group, like for this example. Then we combine our queries, then viewing our result. For example, there are 73% having these two probability of hydrogen bond formation. After that, we enter the program, which is called Mercury, to detect the hydrogen bond length. This Mercury will give us the length of hydrogen bond. After that, the statistical analysis and drawing the histogram will give us an idea about the probability of hydrogen bond formation. For example, if we have a moderate hydrogen bond, it will be between 2.5, 3.2 angstrom, and weak hydrogen bond between 3.2 till 4 angstrom. The stronger hydrogen bond will be below 2.5 angstrom. This is an example from my previous work, from my PhD thesis in the University of Nottingham. So, for example, my drugs was pylodipine, and felodipine made as it's a poorly water, practically insoluble drug. It's a crystalline drug, and it's mixed with other polymer by milling method, cryomilling actually, cryomilling method. So first of all, we build our data according to chemistry structure database. So we select the motif of felodipine, donor and acceptor group from the polymer. Then we detect the length of hydrogen bond. So for felodipine with HPMCAS, felodipine with PMMA, and felodipine with the Solu+, there is a good probability of hydrogen bond formation. For felodipine with HPMC, hydroxypropylmethylcellulose, there is weak probability of hydrogen bond formation. And for felodipine with the Solu+, there is other site for hydrogen bond formation, which make it a strongest solid dispersion. To prove this theory, we made a stability study for recrystallization for 6 months. For example, this is a stability study that was done for felodipine with HPMC and with the Solu+, with HPMC, with these all polymers. Then we detect the recrystallization tendency. It's calculated from the DSC program, and XRPD is additional conformation. So at 0% relative humidity, 25 degrees Celsius, there is a recrystallization of hydroxypropylmethylcellulose with the felodipine. And this is support our idea. And we can see Solu+ hydroxypropylmethylcellulose, there is a weak recrystallization tendency. For PMMA, there is some, there is of course additional factors, but it supports our theory. The big recrystallization was with hydroxypropylmethylcellulose. By increasing the relative humidity, because induction of recrystallization was happened by increasing the relative humidity and increasing the temperature. So here we increase the relative humidity to about 43% degree. We see there is an increasing in the crystallinity of this mixture, especially with the HPMC. And this is of course supporting our idea. And finally, if we increase the relative humidity to about 75% at 25 degrees Celsius, there is more recrystallization happened with the hydroxypropylmethylcellulose. So this supports our theory, and this of course saving more time rather than making this a long and cost stability study test. Anyone can be returned back to this research paper, which is published in 2002 by Ellen. It has 14,000 citations. This is very important article for how to use chemistry structure database and about all statistical analysis that was done. It's very important. So in conclusion, this database will be enable the researcher to select the best drug and polymer with the best hydrogen bond for stable amorphous or co-amorphous system. This will save time and money better than making a long-term stability study to induce recrystallization with the high temperature and high humidity condition. This is, of course, before formulation, and this will be benefit in the formulation study. Thank you for your listening.
Thank you very much, Dr. Ghaida. That was really useful and nice. Now, the next guest speaker in this session, he is an assistant professor in pharmaceutical science. He received his bachelor degree in pharmacy from Jordan University, Science of Technology, and also he received another bachelor degree in computer science with a minor chemistry from Yarmouk University. Then he had his master degree in pharmaceutical science, the University of Texas in Austin. He had his PhD in pharmaceutical science, University of North Carolina. Our next speaker is known for his interest in using molecular modeling software to aid the rational discovery and optimization of lead compounds. using molecular dynamics simulation to provide mechanistic understanding of the signal transduction process and molecular level, and construction of the complete computer software to solve chemoinformatics and bioinformatics problem. Please join me in welcoming Dr. Raed Hassan. Hopefully I'm pronounce your family name correctly, Dr. Raed. Dr. Raed will take us in presenting his topic about computer-aided drug design. You have around 20 minutes, Dr. Raed, and feel free to start. Thank you. Sorry, do we have Dr. Raed or not yet?
Dr. Wright hasn't arrived yet. He'll be joining us in about 20 minutes as per the schedule. Okay, so you may go ahead.
Shall we move with Dr. Cork?
I suggest that you go ahead and receive any questions that you may have for Dr. Ghaida.
Okay, so while we are waiting our Dr. Raed or Dr. Cork to join us, please, if you have any question, raise your hand or write it in the box. We have a congratulation from thanks. Thank you very much. رابط الحضور إن شاء الله راح ينزل. A question about the presentation from Dr. Ghaida. Shall I ask our participant to open the camera, panelists to open the camera so we can take a photo while we are waiting everyone?
Assalamu alaikum.
Wa alaikum salam, Doctor.
Go ahead.
Thanks to Dr. Ghaida for this nice presentation. I want to ask you about if there's any of their students in MSc work on this program nowadays, or
yes.
develop?
Yes, we have actually I have two MSc students, Zulfiqar and Isra, who submitted their paper recently to the journal. We are waiting for the response. Actually, they use the chemistry structure database, and they got benefit from this program. And the new student also that's working now also get benefit from this program and help them in selecting their polymers and the drugs previously before purchasing any polymers. So it's actually it's a prediction study before starting the study, and then if we need to restart our work, then we can add other polymers and then make a prediction.
Wonderful. If there's any of your colleague also use this program with their students in higher education?
Yes, I think Dr. Nidal have a PhD student now. He's working on this program, I think, and also they get a very nice result, I thought.
Wonderful, great. Thank you.
Doctor,
thank you, Doctor. Thank you.
I think there's a question from Amira. Does small molecules such as sugars give a similar effect in amorphization as polymers?
Yes, it can be given if there is a high probability or a good hydrogen bond between the drug and the other additives. As it's high probability of hydrogen bond formation, shorter hydrogen bond will give us a good drug polymer interaction and a stable compound.
Great, thank you.
even we can add other drugs, not only additives, not only sugars or polymers. We can add two drugs together in one formulation.
You mean make like a co-crystal or something like that?
It's co-amorphous.
Co-amorphous, yeah.
Co-amorphous, yes.
Amorphous, yeah. Thank you very much.
Dr. Ghaida, also I encourage you to make a workshop about this program and the college
Of course.
for MS, PhD students and researchers from different universities.
Yes. Of course, doctor.
Okay.
There is a good correlation with the pharmaceutical chemistry department by aiding of this program, a good connection between chemistry and pharmaceutics, and it's very important actually.
Dr. Ghaida, I have another question about application of knowing the hydrogen bond. Only related to the surface of a drug or may happen deeply inside the drug?
Actually, recrystallization may be happen according to the nature of the polymer. If the polymer itself is hygroscopic, it will be allowed to the absorption of water from the atmosphere. According, of course, this will be according to the packages of the drug. Atmospheric humidity will allow recrystallization even inside the formulation.
Right.
Okay. Dr. Ghaida, we have another question from Dr. Mohammed Hassan. Can we use this database to predict the interaction between combination of drugs or just between a drug and polymer?
Between combination of drugs. Sorry. Yes, we can get a good prediction with the small molecules, with the drug, with another drug, or with the drug with the small molecules, such as amino acid. It gives us an indication about the interaction, whatever the additives was.
Thank you. Dr. Ghaida, there is a request if they can get a recorded from your lectures. And I think this is related with the workshop that just Dr. Munder mentioned. So
yes,
if you can share anything
or of course
in the future.
I can write my email now, and then anyone want this presentation, I'll send it to them.
Thank you. Any other questions?
Sorry, Dr. Ghaida. These results are usually in vitro calculations. Do they go in alignment with the in vivo studies?
Of course, if we want to develop or give our work a deepest, more deep information, so we can make an in vivo study to make how much the formulation is stable even inside the body after formulation. So it's important to make the in vivo study. We can make in vivo study and correlate it with the chemistry structure database. We can.
Okay. Dr. Ghaida, we have another question from Dr. Mohammed Hassan. He is asking about the successful rate about this prediction.
Actually, it's even it's previously established in 1963, but they working with this program as not a prediction work. They working from the crystallographic part, from the X-ray diffraction part. But nowadays, from 2002, and actually from Lynn Taylor at 2010, she is the first science, she's from America, that developed this program for predicting the hydrogen bond formation between the drug and polymers. So it's a new we can say it's not a new program, but the predicting of hydrogen bond in the pharmaceutical field is new. But until now, it's very successful. Lynn Taylor made the prediction for a group of drugs with the polymer and for a group of polymer together. And she get a very nice result. And you can get Lynn Taylor a paper from the research gates, and you can see her result. And this is my result from my PhD, is also promising.
Dr. Ghaida, if we follow the question of Dr. An'am, if there is any study about the percent of accuracy between the in vitro and in vivo study?
Until now, there is no study, but it can be, we can make it in our university to be the first study to correlate the in vivo and chemistry structure database.
Okay, thank you.
Thank you, Doctor.
Thank you, Dr. Ghaida. We have a quite positive feedback in the chat box about the promising of this study, the program.
Thank you for feedback. Thank you for all questions.
Any other question?
I think we have a question from Sarah Bedri-Jason. In silico studies usually needed a special computers to run the data. From your experience, can you tell us briefly any significant problems you had during your research?
During my research in the UK, there is no problems, but from the ex-master student of Dr. Munzer, Omar, he helped me for organizing this program inside my computer, and it was very successful. But we should pay for this program. Actually, it's not free. So there is no any problem, and I'll make a workshop to explain how can we use this program, and it's very easy. Maybe can make a prediction for 100 compound within 1 day.
This is amazing, Dr. Ghaida. When we say we have to pay, is it something expensive or is something manageable?
I think Omar said for 20 participants, we paid, each one pay about $500.
4 to 500
for a registration?
For registration per 1 year.
For 1 year, yes. Yes.
Okay.
And really, the students or the one cannot do not need to use a high specified laptop with high quality. Moderate quality may be very enough for application and use of this program and installation this program.
Yes.
So it's friendly in use.
Yes. Yes.
Yeah.
It's easy.
Thank you, Dr. Ghaida. Dr. Ara, do we have our next speaking or?
I'm afraid not yet. I'm afraid not yet.
Okay, so shall we keep going the discussion on? So please, if you have any comments, any feedback, any question, just share it.
Excuse me, just who asked about Dr. Ghaida's address. She's a lecturer in College of Pharmacy, Mustansiriyah University, Department of Pharmaceutics. And she will provide us the lecture, and then we will share too.
Yes, of course. I'm happy.
Let's see.
So, Dr. Ghaida, a question from me: what's the next step from your point of view to deal with this program?
Actually, we are going to make the prediction for hundreds of compounds, and I'm going to make a research area according to the prediction only, and this is of course enough to make a research paper. So I'm going to use this program for a group of compounds, like amides, sulfates, and then see how it's correlate with the drug and how it's correlate with each other, with the polymers. This is my plan actually for this program. And also this program, it's important also to know the type of the polymorph of the drug, because each drugs we have, for example, phylodipine, different polymorph. So if you get and you purchase your phylodipine from one company and you don't know this polymorph, from X-ray diffraction, you send your drug to X-ray diffraction, and then you compare the result from the X-ray diffraction with the data inside a chemistry structure database. Then you can know which polymorph you have, like polymorph 1 or 2. And you know, polymorphism is very important because different polymorph give us different solubility, and also is give amorphization of different polymorph will be different from one polymorph to other. So it's also important for detection of the polymorph type. It's not only the prediction. A lot of usage from this compound is very interesting, and in pharmaceutical chemistry they will get benefit also from this database.
Thank you, Dr. Ghaida.
Excuse me, Dr. Ghaida, I have a question about a crystal structure database. This is like a library for a huge number of compounds. If I want more details about any structure, can I find for XRBD only, or can I find if I want to Raman spectrum, for example, IR spectrum, or just XRBD?
Just powder X-ray diffraction, Dr. Water. Just powder X-ray diffraction. And you can know the melting point also, the crystal shape and form from this compound, not the Raman or infrared. It can be support the infrared spectroscopy in the formation of hydrogen bond. But there is no additional differential scan or Raman spectroscopy was applied in this program.
Right. Thank you very much.
Thank you.
Dr. Raj is around here, so we may expect him to join us in any moment by now.
Welcome, Dr. Raed.
Hi, Dr. Raed.
Morning. Assalamu alaikum.
Wa alaikum assalam. Thanks for joining us, Dr. Raed. I did introduce you, but I don't mind introduce you again. Dr. Raed has received his bachelor degree in pharmacy, Georgian University of Science and Technology. Also, he received another bachelor degree in computer science and minor chemistry at Yarmouk University. So we have a science lover. Then he got his master degree in pharmaceutical science, the University of Texas in Austin. Had his PhD degree in pharmaceutical science, University of North Carolina. He is quite interested in a few areas in using molecular modeling software to aid in the rational discovery of and optimization lead compounds, using molecular dynamic simulation to provide mechanistic understanding of the signal transduction process and at molecule level, constructions construction of component computer software to solve chemoinformatics and bioinformatics problem. I will leave the mic with you. You have around 20 minutes. Feel free to start. Thank you.
Thank you so much, Dr. Samra, and thank you, everyone. Good evening, everyone. It's my honor to be at Mustansiriyah, invited at Mustansiriyah University, one of the oldest universities in the world. Today, I'll talk about computer-aided drug design. Should be able to share my screen. Okay, so today I'll give an overview about computer-aided drug design. I'll start by talking about the drug development cycle, and then the types of computer-aided drug design tools used, such as ligand-based drug design and structure-based, and then a brief overview for the future of the computer-aided drug design. To remind you, you probably have seen this already. The drug discovery or drug development cycle starts with identifying a disease, okay? And then after understanding the mechanism of the disease, you want to identify a target, a drug target that would be crucial for the disease. And then you could verify the target by doing some testing procedures and bioassays. And then after that, you could start looking for a lead molecule or a lead compound that would work on this target. And this phase here is called the discovery phase. Okay? And then you also have the design phase. After you have identified a lead molecule, then your goal is to study the relationship between the structure of this lead molecule and its activity. And this is called SAR. You probably heard of that. And then you would identify the parts of the molecule that are essential for activity, which are known as pharmacophore. And then your goal after that is to optimize this lead by enhancing the pharmacodynamic and pharmacokinetic properties, okay? And this phase here is called the drug design phase. And then finally, you have the preclinical and clinical phases. In the preclinical, you would check the selectivity and toxicity of the molecule, make sure it has no side effects. And then you would study its metabolites, and then stability of the drugs, make sure that it's chemically stable. And then you would also, you would also check the manufacturing process. Some drugs might be difficult to manufacture, others might be easier. So this is one of the processes also involved. And then finally, patenting and the regulation affairs of the drug. And then of course, there is phase 1, 2, and 3, testing the drugs on human up until the last phase of the post-marketing. Now the computer-aided drug design could help mostly with finding the lead, as we'll see today, as well as identifying the pharmacophore and pharmacodynamic properties. These are the main uses for computer-aided drug design, but they could also be used to optimize pharmacokinetics. They could also be used to predict the selectivity and toxicity. There's also some software that could actually predict the metabolites and the stability of the drug manufacturing process, okay? But the most ones that are most commonly used for computer-aided tools, one of the goals used by computer-aided drug design are actually finding the lead and identifying the pharmacophore and optimizing the pharmacodynamics. Okay, so what is computer-aided drug design? After you identify the target, then you will have two scenarios. The first scenario is in the case where you know the target, but you do not know anything about the structure of this target, okay? So let's say you have a number of active molecules. You know they are active, but you have no idea about the structure of the target or how these molecules bind to the target. And so in this case, we'd use something called ligand-based drug design. So the design is based on your knowledge of the ligands or the molecules that you know already they are active, okay? On the second scenario is if you have the structure of the target, you do know the structure of the target receptor, then you would use something called structure-based drug design. So we'll have ligand-based drug design and structure-based drug design depending on your knowledge, okay? Now for ligand-based drug design, we have two tools. The first one is called Qsr, as we'll explain. Another one is called Pharmaco Model. In the structure-based drug design, we also have two commonly used tools. One is called de novo drug design, another is called docking. Okay. Now, something I want to mention, sometimes you may not know the target, the target receptor, but you may know the primary amino acid sequence of the receptor, the protein. In that case, you could actually use some modeling tool, such as homology modeling, protein threading, recently developed methods such as AlphaTo and Rosetta Fold. You could use these tools to predict the structure computationally, okay? And then after you predict the structure, then you will be able to use the structure-based drug design. Okay, all right, so let's start with the first type of methods, which are the ligand-based drug design. Okay, so the first one in the ligand-based drug design is the Qsr. Right. So, like we said, in the ligand-based drug design, you have a number of ligands that you know they're active, but you're really not sure how they bind to the receptor. Okay, so if you have, for example, a number of these compounds and you know some activity for them, so then you would computationally generate something called the physicochemical properties. Okay, so these are physicochemical properties for the drug. For example, you might calculate the surface area, the polarity of the molecule, the number of oxygen molecules in the molecule, and so forth. You would create all these, which we call physicochemical properties or descriptors, okay? And your goal is to relate the activity to these physicochemical properties and come up with an equation like this. So your goal is to come up with a function that identifies the activity, that relates the activity to these physicochemical properties. So activity would be a function of the x1 to until m. Okay, this is called the Qsr equation. Okay, so once you identify the Qsr equation, you could actually apply it to search for new molecules. See, in the case with the ligand-based drug design, you have a small data set of molecules that you know they are active, but your goal is to invent or create new leads, new molecules that could also be active. So you want to build an equation, Qsr equation, and then apply it to a database of molecules to search for new molecules, okay? So here is an example of applying the Qsr equation. We would usually have to screen a database of molecules, let's say 1 billion molecules, for example. And then these are a database of molecules that we want to dig for and find if it has any active molecules. And then you would apply the Qsr equation and predict the activity. Some of the molecules will be, of course, active, be identified as actives, while others might be identified as inactive. So those that are identified as active could be further tested experimentally. Okay, so that's all about Qsr. All right, so now another method that is also a ligand-based drug design method. Another method that is a ligand-based drug design is called the Pharmaco Model. Okay, so again, we have a data set of molecules that we know they are active, and our goal is to identify something called Pharmaco Model that could be used to screen databases and find more active molecules. So first of all, as you may know, definition of the Pharmacophore are a set of structural features in active molecules that are recognized by the receptor and responsible for the activity. Examples of Pharmacophore structures, pharmacophoric features are the hydrogen bond, acceptor and hydrogen donor, positively and negatively charged groups, hydrophobic region. Okay, and then what's important is after you identify these features, you also want to identify the relationship between them. So you want to identify the relative position, the relative relationship, or how they are oriented in space to complete the definition of the pharmacophore. So, for example, this one here shows a full pharmacophore model for a data set of molecules. Notice that you would identify regions in the molecule by their physicochemical feature, such as negative charge, hydrogen bond acceptor, hydrogen bond donor, and so forth. So hydrophobic region, hydrogen bond donor, and then you would also identify the distances between them, because just having these groups alone does not guarantee that the molecule would be active. It has to be oriented in certain region to be able to be active and bind to the receptor. Okay. So let's look at an example. Here's some active molecules, you know they are active. And then your goal is to identify the common pharmacophoric groups among them. So first you would generate all the possible configurations in the molecule. So for each one, you would generate some conformations, okay? And then you would overlap these molecules with all the possible scenarios to identify the region where to identify the common regions that could identify the pharmacophore. So in this case, for example, you would see that this region is common, this region here is common, and then this is this one here and this one here. So you would assume that these groups are, since they are common, you would assume that they are essential function groups, and then hypothesize that this is a pharmacophoric model. Okay, and so again, just like we did with the Qsr, you have a model here instead of the Qsr equation, and then here is your database where you would search for molecules that are possibly active by applying the model. So if any of these molecules have, you know, the pharmacophoric models identified, if it fits the model, then they would be identified as active. If not, they would be inactive. Okay, so that's as far as the ligand-based. So far we covered Qsr and pharmacophoric model, which are the ligand-based drug design. Next, we talk about the structure-based drug design. Okay, like we said, in this case, we do know the structure of the receptor. Okay, so we could use de novo or docking. I wanted to say that one of the most common tools in the ligand-based is actually pharmacophore modeling. That's why it's highlighted in red. It's more common than Qsr. And then, as far as structure-based drug design, the most common one used is docking. It's more common than de novo, as I'll explain why shortly. Okay, so let's start with the de novo. We know the structure of the receptor, so that's the receptor here. Okay, and we know some binding regions here. Okay, for the receptor. And we're going to build molecule from scratch. So de novo means you're creating a molecule from scratch, from zero, to come up with something that would bind and interact with the receptor. So you would start with putting some function groups, or we call fragments, at the regions that interacting with the receptor, and then you would bridge them, connect them together, and then come up with a new molecule. Okay. There is a software called Ludi I will use as an example. So let's start with the binding site of the receptor. You would identify the interactions, and then you would add fragments to these binding regions, and then you would connect them, bridge them with some groups, with some linkers. So here are some examples of the molecular fragments used, and then the bridges used. So what's unique about de novo is that it come up with molecules that they've never been designed before. So that would guarantee you that you could come up with something new that you could patent. The problem with de novo is sometimes you may come up with a molecule that might be impossible to synthesize chemically, or sometimes you might come up with a molecule that we are not sure if it's going to be safe or toxic. So there is risk in developing molecules that we've never seen before. Unlike the previous cases, when you have a database of molecules that we know that they are safe, we know how to synthesize them, we know everything about them, we just have to screen and find something that is active. So that's the major drawback or problem with de novo. Okay, right. So the second method in structure-based drug design is something called docking. So in docking, you would place the molecule, search for molecules that would fit the binding sites. Okay. So it's a computational simulation method to place the ligand into the receptor. And you have something called binding mode, which is the way the drug binds to the receptor. That's called the mode. Okay. And then finally, you have a score. So to measure how good the fit or the binding is, you would have to calculate something called score, or we call as scoring function, referred to as scoring function, okay? So these are some definitions for docking. And how do we apply docking? So you would have a database, just like we've seen before, you have a database of molecules, and then you would perform docking to the receptor. You know, there is the structure of the receptor that's known already. You would have to take each molecule and dock it into the receptor, score it, measure how good the binding is, how good the binding is. And then if it has a good fit, then you would assume that it could potentially be a good drug. If not, you would throw it away. So that's one of the applications of docking. Another application of docking, sometimes you know that this drug is active, you know that it binds to the receptor, but you are not able to experimentally see how the drug binds to the receptor. Usually you would use X-ray, NMR to see how the drug binds to the receptor, but if you're not able to do that for maybe cost reasons, or maybe sometimes experimentally, you might not be able to generate a good crystal to do the X-ray. So in this case, you could use modeling, you could use docking to see how the drug binds to the receptor. So you would try to best guess using modeling to fit the drug into the binding site, okay? And after that, you could actually use what's called lead optimization. You could look at how the drug is interacting with the receptor. Okay, so this is the receptor here. These are all parts of the receptor. So your goal is to optimize the drug such that it's interacting more with the receptor. You could add function groups to enhance the binding. So these are the main applications of docking. Now, docking methods can be classified into three major ones. The first one is when you have a rigid receptor, you're not making any changes to it, and a rigid ligand, and then you try to find all the possible ways how the drug binds to the receptor. The second way is when you have flexible ligand, but rigid targets. So the ligand could be flexible. You could try to, generate all the possible conformations and dock it. Okay, and the third way is when you have flexible ligand and flexible receptor. So both ligand and receptor could be flexible. So the most accurate one is, of course, when you have flexible ligand and flexible receptor, but this is expensive in terms of computer time, okay? The quickest and least accurate method is when you use rigid target and rigid ligand, and somewhere in between, which is when you have rigid target and flexible ligand. This is the most feasible, has some accuracy, but it's tolerable. You could use like personal computers to perform this docking. Okay, so that's the most common one. How do we measure the fit? How do we measure how good the binding is? We use something called scoring functions. So with scoring functions, could be classified into three different kinds. For the sake of time, I'm just gonna skim through them really quick. So the first one uses some physical forces, using Coulomb interactions and Lennard-Jones equations to measure how the interactions between the charges. The second one is empirical, which means you compare your results with some experimental data and see how comparable they are. And finally, you have knowledge-based, which means you mine or extract some common way of how the drug interacts with the receptor, and then use that to measure the score. Now, usually to get the best results, you use something called consensus. So you would have multiple forms of scoring functions, and you would use an average for all these scores. Am I okay with time? Okay. So the last.
Yes, please keep going.
Okay, good. All right. So the last thing I want to talk about is the future of CAD. Where are we going with this field? I think this is important because if we want to catch up with where people are at, it's better to look for what they are going to instead of trying to reach what people have achieved so far. Dr. Ahmed Zuhail, the Nobel Prize winner, once said that if we want to catch up of where people are at right now, we should be looking for where they are going, not what they are at this point. Why is that? Because if we try to catch up what people are right now, by the time we reach there, they would have reached to another level and advanced to another level. So our goal is to see where they are going and then try to achieve that. So at some point we will be both at the same level. Okay, so it is important to see where people are going with CAD and then try to go to the same direction instead of trying to achieve what people have achieved so far. Does that make sense, guys? Okay. So where are we going with CAD? Right now, everybody, CAD or the computer-aided drug design is getting a lot of interest nowadays, especially with the development of many machine learning and data science tools. There's a lot of AI, artificial intelligence, and machine learning tools that are now being successful and used successfully in many fields, including designing drugs. And so applying these machine learning tools has proved a lot of usefulness. So for example, there's been a successful method called AlphaFold 2 and RosettaFold that uses AI and machine learning to predict the protein structure. So we should be looking at these methods as ways to enhance computer-aided drug design. Another thing that's been advancing also is using MD simulation to predict binding affinities. Predicting how the drug binds to the receptor using these rigid and flexible methods are not the most accurate. MD simulation is more accurate in making these predictions. And MD simulation also, the problem with MD simulation is it requires a lot of computation time. So there's been a lot of advancement in the computers, the supercomputers themselves, okay? And then finally, CAD is also interested into finding a comprehensive approach where you could actually make all predictions at once. And instead of just looking at how the drug binds to the receptor, right now, the field is interested into finding a comprehensive tool that could predict everything from A to Z, from the time the drug binds to the receptor to the pharmacokinetic properties to toxicity and everything. So find a comprehensive approach. So if you're interested in this field, I think you should be catching up on all these future developments, directions of CAD to be on the top of the field. Thank you so much for your time again. It's an honor to be with you guys in Baghdad and in Mustansiriyah. If you have any questions, if I have time, I think I'll be glad to answer them. Do you have time for, do you usually have question answer session, or is it just lecturing only? I'm sorry, Amra.
Thank you, Dr. Raed, for this very unique presentation. It's very quite interesting, and I think we need more than 20 minutes for this presentation, but for time consideration, we will leave the question for the end of the session. If we have our next speaker, Dr. Peter Cork. Dr. Ara, would you please confirm, do we have our next speaker?
Dr. Peter is around. I'm not sure if he's ready. He's got 10 minutes yet, but he's joined us already, so he can hear us.
Okay. Yes. So
yes, I can hear you.
Hi, Dr. Peter. How are you?
Good.
Thanks for joining us. So our next speaker is Dr. Peter Cork. He received his bachelor degree with honor first class in pharmacy in 1966. He finished Master of Science in Pharmacy in 1967, had his PhD in Pharmacy and Doctor of Science in all of University of Manchester, UK. Very impressive. He was a faculty member in Department of Pharmacy, University of Manchester from 1969 to 81. Dr. Peter also spent 2 years as a research associate in the Department of Pharmacology, and he is internationally recognized for his research and work in anti-cancer drug discovery and delivery and development. He leads a research team to discover and developing new cancer treatment drugs with an emphasis on lymphoma and leukemia. He also has an active program in drug abuse, which focuses on the development of a new treatment for nicotine and alcohol addiction. He graduated 30 PhD students, 9 master degrees students, and trained over 60 postdoctoral fellows and visiting science. He also established a co-founded 6 drug discovery companies. Congratulations on that, doctor. And his drug discovery patent and application total more than 270. At present, several of his laboratory drugs are in different phases of clinical development. During his career, Dr. Cork has authored more than 600 pre-reviewed research articles and 700 symposium abstracts. Thank you very much. You have 20 minutes, Dr. Peter. Shall I ask you to share your screen, please?
Yeah, so I'm not sure how I do that. Let's see.
So maybe Dr. Ara will give you some tips.
Well, there's that green button in the middle, the screen, share screen, it says. You usually find it below at the toolbar.
Oh, window. Okay, I think that is that okay?
Let's cross our fingers.
Not sure what you're seeing.
Not yet, nothing yet.
Having problem sharing screen.
Oh, you don't.
That's all right. Technical issue always happens.
Can I see your mouse for a second? Let's do. Exit this. Where's the Zoom? So click on your.
There you go.
Okay, I think let's put it on presentation mode. Will it go on presentation mode?
Should, yeah.
Oh, well down there. I don't want this screen here.
So what do you want? What do you want here?
I want that one on again.
Okay.
Okay.
Perfect.
I think you're okay.
Yep.
Should be.
Okay, I think we're on. All right now. Okay, well, thank you for the invite to talk to your virtual conference. I enjoyed the previous presentation by Dr. Kashan, and what I thought I would do is talk about a recent project that we've been involved in, involving some sesquiterpene lactone derivatives, natural products that we're trying to develop analogs for to get them into phase 1 studies. And it turns out that the targets are quite interesting and complex, and we've made a lot of progress in this area by using molecular modeling. So it's a really good example of building on what the previous speakers talked about, and how you actually use those techniques in the practice of drug discovery and design. So we've been interested in the sesquiterpene lactones for some time. We're particularly interested in the ones you see on the screen. They're related to parthenolide, a very well-known sesquiterpene. We have had an interest in 2 more of these natural products, melampomygalide B, which you see structure in the middle, And another one, meshelialide, this is recently just entered in development and probably will get into phase 1 studies soon. Parthenolide has been in clinic, but it failed due to problems in bioavailability. And melampomagnoloide B is the one that we're particularly focusing on, and we'll tell you the story about that one as we go along. You can see that all these natural products are from a number of plants. Parthenolide from Tanacetum parthenium, melampomagnoloide B, as from the name, from the magnolia tree, Magnolia grandiflora, and the meshelialide from a plant that grows in Africa, Michelia champaca. We're going to focus on parthenolide and melampomagnoloide B, which we abbreviate MMB. So we know that you can't protect natural products because it's very difficult to get composition of matter of known natural products. So what we plan to do is two things. We make novel molecules that we can protect, and we also design them in such a way that they have good drug-like properties and are designed to target proteins of interest. So we were interested in parthenolide. I wonder if I can get rid of that. Nope, I can't. Because it had an interesting profile against leukemia cells, and was initially thought to be a promising drug to treat various forms of leukemia. We were particularly interested in acute myelogenous leukemia, and you can see the data on this slide shows that parthenolide does very well against AML stem cells. And if you look at the effect on in the dose response, this is viable cells, you can see that over the concentration range of 1 micromolar to 10 micromolar, you see a really good effect on cell viability with parthenolide. PTL is the abbreviation for parthenolide. And when you look at normal bone marrow stem cells, there's virtually no effect on normal bone marrow stem cells. So you have some really good selectivity there for the leukemia stem cells. And when we compare it with a standard drug that is used to treat leukemia, this is acyclovir, RSC, we don't see really much effect at all on the viable AML stem cells. And this is one of the big advantages of parthenolide, that it attacks stem cells. And that's really a key to developing a drug as an anti-leukemic agent. And you'll see on the right-hand side, what I've done is give you an indication of how these molecules work. In this mouse leukemia model, not sure if I can get rid of that. There we go. You can see that in a leukemia mouse model, and take these models through grafting patients' leukemia cells, AML cells, into the bone marrow of the nude mice. They're immunocompromised, and so they take and they actually produce AML stem cells, the human AML stem cells in the animal, and then we can treat the animal with various drugs to see whether, in fact, these drugs are effective. And we can also look at certain biomarkers to determine what particular proteins are involved in the mechanism of action of the drug. And in this particular case, we see that as you increase the dose of parthenolide and look at the progress of the blood samples over a 6-hour period, this is giving a massive dose of an analog of parthenolide, DMAP, 150 milligrams per kilogram. We see a definite effect on a protein called NF-kappa B, which is an anti-apoptotic protein, and that we'll talk about in a little while. But with this massive single dose over this 6-hour period, you see a complete absence at the 2-hour marker point of this particular biomarker. And this gave us a pretty good indication of where these molecules are interacting. So we wanted to make sure that we weren't missing anything in the mode of action because some of these sesquiterpenes sometimes interact on multiple targets. And so we decided to make an MMB biotin probe. And this is the structure that you see in A. It has a biotin connected to the sesquiterpene lactone via a long linker. And then we did a streptavidin pull-down experiment where we treated the AML cells with the biotin probe and then looked for the binding of this probe to the proteins of interest and carried out 2 analyses. One was Western blot analyses with various antibodies to determine the identity of the proteins. And the second one, you see the LCMS identification, was doing a proteomic analysis by identifying the proteins through LCMS sequencing. And doing it this way in parallel, we got some very strong evidence that the proteins of interest kind of fitted what we thought. This IKKB target here that we see, which is a valid target, you see it turns up in the biotin probe, and it competes very well with parthenolide in the competition assay. It competes out, so that's a valid target, and it's shown both in the competitive binding assay and the LCMS assay. Further down, we can see a lot of these other proteins, the ones that have the G in front of them. These are all elements of the glutathione pathway, and they're all really critical proteins in the synthesis and catabolism of glutathione. So we knew straight away that this particular sesquiterpene had 2 major modes of action. One was on the NF-kappa B pathway because IKK beta is a really pivotal component of that pathway. And then we had a number of elements of the glutathione pathway as well. And we considered these 2 pathways as being synergistic. And so it certainly appeared to be a very interesting molecule in that you had 2 different modes of action that were complementary to each other and likely synergistic in the anti-cancer mechanism. So in the glutathione story, you saw that there were a number of glutathione pathway proteins that were pulled down by the streptavidin experiment. And I've shown here this rather complicated pathway of how glutathione is synthesized. You see in this yellow arrow that's going through a number of intermediates, that this is the pathway that biosynthesizes glutathione, and then glutathione is involved in a number of other interactions, basically to try to destroy reactive oxygen species that can be very damaging to the cell. And this involves glutathione transferases, GSTSs, glutathione peroxidases, reductases, and this unusual one on the membrane, the thyroidoxin, which is another molecule that gets rid of reactive oxygen species. And you see all the black arrows are where these sesquiterpenes interact. And so you can actually prevent glutathione metabolism in addition to preventing its action in reducing reactive oxygen species. So now we know that these sesquiterpenes have a dual mechanism of action. One is that they inhibit NF-kappa B through inhibition of IKK beta, which is a kinase enzyme, and they also inhibit glutathione function and biosynthesis. If we look at the NF-kappa B inhibition, you'll see the IKK beta that pulled down in the streptavidin experiment is that red square there, which is one of a component of 4 IKK units, IKK alpha, beta, and gamma. There are 2 gammas, which are usually called NEMO, and there's an alpha and a beta. The alpha has a relatively poor kinase, but the IKK beta, the IKK beta is a kinase that is able to function as a phosphorylator. Yeah, I got somebody talking on the line there.
Sorry. Could you please mute yourself?
So I want to go over this pathway because it is an important one when it comes to the modeling. The kinase complex that has these 4 units is very efficient in phosphorylating this component that the arrow is pointing to, which is a component that contains a protein called I-kappa B, which is an inhibitor molecule, and 2 protein units called P50 and P65, which constitute NF-kappa B. And the normal mechanism is that this kinase phosphorylates this inhibitory complex in the cytoplasm. And then once the phosphorylation occurs, it breaks down into NF-kappa B, which is this P50, P65 dimer. It contains a phosphate group that has been put on by this IKK beta kinase. And then the inhibitory part of that complex, I-kappa B, dissociates and is then ubiquitinated and then interacts with a proteasome and gets digested and destroyed. And then the released active NF-kappa B is then able to pass through the nuclear membrane and interact with the anti-apoptotic sites on DNA. And what this does, it stops the apoptosis of the cells, if these are cancer cells, and allows them to proliferate. So what cancer cells do is they chaperone this particular pathway and prevent the destruction of their own cells through this normal apoptotic pathway. Now, in the second mechanism, we managed to get some information from molecular modeling, which is quite interesting. We've looked at the 2 major components that are involved in the glutathione mechanism. One of these proteins is a protein involved in biosynthesis of glutathione, the GCL enzyme. This is the glutamyl cysteine ligase, and it's an interesting enzyme because it's a component is a catalytic part called the GCLC catalytic moiety, which you see, this is the larger unit that you see. And then the smaller unit is the modulatory protein unit, which is needed to activate the catalytic protein. And there is an interaction. If you see the modeling here, you can see 2 cysteine residues, cysteine 193, which is in the modulatory part of the complex, and then cysteine 553. And in the mechanism of this enzyme, these two cysteine residues come together, they form a sulfur-sulfur bridge, and that causes activation of the catalytic moiety. And the active site is on that GCLC unit. You can see that parthenolide is interacting there. That's a red molecule there that you see. And also you can see that parthenolide interacts right at the interface of these two cysteine residues, which join together the catalytic moiety and the modulatory moiety. And what we found by using the Schrödinger molecular modeling suite is that the interaction of parthenolide with cysteine 553 and cysteine 193 is one that involves a covalent binding of parthenolide to that 193 cysteine, and that prevents the formation of the disulfide bond between the catalytic unit and the modulatory unit, and therefore blocks the activation of the enzyme in forming the product in the penultimate step on the way to the biosynthesis of glutathione. You can see the Western blot data below. The parthenolide is very effective in blocking this particular enzyme in a number of AML cells from patients, clinical samples, and also the immortal cell line M9-ENL. Sometimes you get some cells that are resistant. For example, AML10 is resistant to inhibition of GPX1, the peroxidase enzyme, when treated with parthenolide. But the GCLC looks like a pretty good target there. The effect over 6 hours of a single 7.5 micromolar dose of parthenolide looks pretty good. So these are the two mechanisms that are synergistic, and you can see why on this chart here that both of them lead to apoptosis, and both of them are different mechanisms that reinforce each other. What we were interested in doing was trying to stop this ability of the cancer cell to prevent this pathway from targeting its cells for death by blocking this important kinase, and also trying to increase the reactive oxygen species within the cancer cell, so that the two combined mechanisms would kill the cancer cell. And we know from previous work that cancer cells are quite deficient in active glutathione. So any molecule that reduces active glutathione in the cancer cell is really going to work very well, because normal cells usually have a good supply of active glutathione, and they probably will not be affected as much by parthenolide in the glutathione pathway than cancer cells would, that have very little reserve of active glutathione. So one thing I would like to say, just in passing, and we'll pass through that fairly quickly, is that we have made this interesting amino analog of parthenolide, which is shown on the right here. It's called DMAP, and it's made very simply from parthenolide by a Michael addition of a dimethylamine across that exocyclic double bond. And what this does, it actually forms a water-soluble analog of parthenolide. And of course, water solubility is a big problem, and that's why parthenolide failed in clinical trial. But DMAP is about 1,000 times more water-soluble. It has a 75% oral bioavailability compared to about 1 or 2% oral bioavailability of parthenolide. And it, most importantly, is equipotent with parthenolide against AML stem cells, and has a similar kind of mechanism, somewhat different though, as we'll see later on. Its pharmacokinetics is shown in this table, and it looks pretty good in these three species, mouse, rat, and dog. both IV and PO. PO looks pretty good. The bioavailability, as I say, is around 65, 75%. And these were designed as water-soluble prodrugs. We believed when we designed these molecules that DMAP would convert back to parthenolide in vivo through a reverse Michael addition, where this reaction that you see here goes the opposite way, and you release dimethylamine to form parthenolide in vivo. Turns out that that wasn't the case, because we only see about 2% of parthenolide formed in vivo. And so these amino analogs are not really water-soluble prodrugs at all. They're stable, fairly stable molecules in their own way. So in the clinical trial, there's just one thing, well, a couple of things I wanted to mention. We were able to dose patients with DMAP, and we're also able to see this biomarker NF-kappa B that we saw in the earlier study in mice. We saw it in humans in the blood samples, and we're able to see it disappear on daily treatment of the patient with parthenolide, DMAP, and this was a single bolus, a 20 milligram per kilogram dose over a period of 28 days, and we're looking at this. There are two boluses there, one over the first 28 days, and you can see there's a little bit of delay before the NF-kappa B is starting to disappear. And then another 28 days reinforces that and keeps that NF-kappa B very low concentration. So we know from this biomarker that the drug is working. Now let's go into the NF-kappa B mechanism, because that's the one I want to really focus on, and it's really a complicated target. This is some work from a Poli publication back in 2013, where they managed to get a crystal structure of the IKK beta protein. And you see this in different orientations of the molecule, and you can see the various interfaces that we'll talk about in a little while. The most important thing to notice is that when you look at how the molecule is put together in its active form, it involves a oligomerization. And so the major form that we see is the dimer. The monomer is also there, but it doesn't have much activity. But is the cartoon of the dimer. We call this the closed form of the dimer. And it has certain interfaces on it that are really important for activity as we move to the active form, which is an oligomer of the monomer. And we see three areas, a KD area, which is the kinase domain, a ULD area, which is the ubiquitin-like domain, and then a scaffold dimerization domain, which is a rather large domain here. And these all have important contributions to the activity. If you carry out mutations in any of these three sites, then it definitely affects activity in a large way. But the kinase domain is the most important one that we considered, because we wanted to block that site so that we could stop phosphorylation of the activation of NF-kappa B. But in this process, what happens is the closed dimer here that is formed opens up by a conformational process. And then once it opens up, it's able to oligomerize into open dimers, tetramers, and eventually oligomers that are octamers. I think octamers is the optimal form. And interestingly, they come together in a way that is unique, in that all the KD sites are connected together in a cyclic form. And this is a way that the molecule phosphorylates the substrate, which is the inactivated NF-kappa B complex, through a trans-autophosphorylation loop that accelerates the phosphorylation and allows very rapid phosphorylation of the substrate. As we move from dimer to octamer, then the kinase activity increases dramatically. The dimer has very little kinase activity, but as it oligomerizes, then the activity increases. But we were interested in looking at this whole process and how these parthenolide analogs might interact with this very complicated oligomeric system in a way that we could control phosphorylation of the NF-kappa B complex in the cytoplasm. So looking at this a little further, if you look at more detailed analysis of the monomer, we can see it's made up of the kinase domain, the kinase domain, which is the green part. The ubiquitin-like domain is in the middle there, and then you've got this very large area, helical area, which is the scaffold dimerization domain. And all these are sites that are really interesting ones that one could look at, which might result in modulation of the phosphorylation of the I-kappa B complex. Now, in this diagram here, we don't see the part on the end, which is the NEMO part. That's the one that contains the amino acid sequence that binds the IKK gamma units. There are two of these units, and the crystal structure doesn't contain that. This is human IKK beta. What we did is we remodeled this by annealing the NEMO binding area, and that constituted the first time that the whole sequence had been computated into a molecule that we could utilize in looking at small molecule interactions. So a lot of chemistry, of course, is needed to carry out these structure activity studies, and we carried out most of our synthetic efforts on this molecule MMB, which is a close relative to parthenolide. It's useful because it has an hydroxyl group, and then we can functionalize it and make a host of different functionalized analogs, and then use these models that we just talked about to determine whether we can see any interaction at these interfaces that we believe are good targets for shutting down this phosphorylation of the I-kappa B complex. We were particularly interested in this one here, 7H, when you can see from this National Cancer Institute screening data that it does very well against a whole range of different human cancer cell lines. Not only does it work very well against leukemia cell lines, but it also works against a whole range of solid tumor cell lines too. And you can see some of the data here in these different patterns. There's breast cancer, there's colon cancer, and there's also renal cancer, where it does extremely well, in addition to its activity in the against leukemia cells. So we were very pleased to see that this particular molecule had a wide range of activity against both blood cancers and solid tumor cells. And looking at the interaction of these molecules with the monomeric form, we wanted to focus on the monomeric form. Obviously, it was difficult to attempt to model the octamer, which is the more active form, but we hypothesized that if we could find molecules that interact with the monomer, then we may be able to block not only phosphorylation, but also oligomerization, and also some of these other areas like the NEMO binding area that is now part of this molecule. Now this is the complete structure of the IKK beta monomer with the NEMO binding unit now incorporated. And we found that these click chemistry products that we developed, in particular 7H, this is the one I showed you the data on, interacted with the kinase domain, and you can see this is the kinase domain area down here. Here's the ubiquitin-like site in the middle, and then the NEMO binding site is the part that was annealed onto the crystal structure. And this is the SDD site, rather large site, and it really extends down into the area that's close to the kinase domain. And this is where we found that this molecule number 10, which we wanted to look at very, very carefully because it was like DMAP. It was a Michael addition product with dimethylamine, which converted 7H into a water-soluble and more drug-like analog. And we knew from our parthenolide and DMAP studies that these molecules really should behave the same, but 10 should be much more drug-like than 7H because of the presence of that tertiary amine in the molecule that would give it water solubility. Interestingly, what we found was that unlike DMAP and parthenolide that were thought to interact in the same way with this dimer, this monomer, they interact at different sites. When you put the amino group on 7H to form 10, then you got a switch from interaction of the KD domain with 7H to the bottom part of the scaffold dimerization domain, which you can see here. This is right at the bottom there. And that amine causes a shift in the way that these molecules bind. And so we thought that these probably would work by different mechanisms. 7H is very likely a competitive inhibitor at the kinase domain, but 10 is likely an inhibitor of the dimerization and subsequent oligomerization of the monomer because it's interacting with that really important site at the base of the scaffold dimerization domain, which is involved in that opening of the dimer, if you remember, that allows it to oligomerize further to the tetramer and octamer. And you can see that on the next slide, you can see a better idea of how these things are working. So DMAP is shown here, and it's interacting at exactly the same site as 10. DMAP has that amino group in its structure, as we saw earlier. And so both DMAP and compound 10 are amino analogs of the sesquiterpene lactone, whereas MMB, we saw MMB before, and parthenolide and 7H still have that exocyclic double bond, and they interact very similarly at the kinase domain. So you can separate out these two forms, and they're probably acting by different mechanisms. The MMB PTL 7H trio are interacting at the KD side and are likely to be competitive antagonists of the phosphorylation event, whereas DMAP and compound 10 are likely interfering with the dimerization and subsequent oligomerization of the monomer to its more complex tetramer and octamer forms. So we wanted to find out if 7H really was interacting with the KD site and interfering with phosphorylation of the I kappa B complex. And if you remember, the I kappa B complex contains NF kappa B in a dimer form P65, P50, and the P65 is the unit that's phosphorylated by IKK beta. So in this Western blot data, you can see the phosphorylation event. The first part is parthenolide as a positive control, and we can see that as you increase the concentration of parthenolide, you see a decrease in the phosphorylation of P65 in the I kappa B P65, P50 complex. That's the inactivated complex of NF kappa B. And when you compare it with 7H, you can see that 7H over this concentration range is more potent than parthenolide, but it does inhibit significantly the phosphorylation of P65. So the modeling fits the actual data that we are seeing an interaction with the KD site, and there's an inhibition of phosphorylation of the IKK beta kinase in its ability to phosphorylate the I kappa B P65 P50 complex, which is the NF kappa B in its inactivated form. And here you'll see some data on the cytotoxicity. These are breast cancer cell lines, and you can see with I can't see that one because of the blue line is a cell line, a human cell line in the NCI screening data unit. And you can see that that's a pretty potent inhibition that we see with compound 7H, far more potent than you would see with parthenolide. And the other two have a little less potency, but are still pretty potent compounds when you look at cell survival of these two breast cancer cell line forms. Here we looked at the ability to inhibit the binding of NF kappa B that is released from the inhibitory complex with DNA. This is you can see a concentration effect of 7H. Here we're seeing binding of NF kappa B to DNA, and the reason why you see this concentration effect is that there's less NF kappa B being formed in the cytoplasm because 7H is a very good inhibitor of the I kappa B phosphorylation that releases NF kappa B. And so you're seeing less active NF kappa B passing through the nuclear membrane and interacting with those apoptotic sites on DNA. At the bottom, we see an interesting one where you see TNF alpha. TNF alpha is an activator of the NF kappa B pathway because it cycles in releasing active NF kappa B. And you can see this, that if you have no drug present and you monitor the effect of an interaction of TNF alpha in the cell, you see over a 60-minute period a disappearance of I kappa B, which is the inactivator of the NF kappa B complex. And then you see that disappear. That's a cycling at the 60-minute period. So we postulate that if this cycling is inhibited, that we should see less IKK B alpha, I kappa B alpha, in the cytoplasm being activated to NF kappa B. And this is exactly what we see, that with the drug present, you can see I kappa B alpha is still there, relatively not changed. There's a little bit changed at the 30-minute point, but not much at all. So NF kappa B is being kept in the inactive form in cytoplasm when the drug is present. But when the drug is absent, the activated TNF alpha is causing the NF kappa B to be converted into its active form, where it can bind to DNA. This fits this upper study very well. So we think we know a lot now about how these molecules work in the cytoplasm. And here's a good schematic of where we see these molecules interact. So this is the monomeric form of IKK beta. We see the four areas, the kinase domain, 7H parthenolide and MMB interact with that site. The ULD domain, we don't have any interaction there, but we do have interaction at the STD site where the amino parthenolides interact. And they interact at the base somewhere down here. And this is where this base is very important. When it opens up into the open dimer form, it's likely that compound 10 and DMAP are interacting with the base of this STD area and maybe preventing the opening up of the dimer and subsequent oligomerization. We're looking at that to see whether we can figure out a way of determining if that's a correct hypothesis. But the main event, the inhibition of KD would mean that you would block both the oligomerization and the ability of the molecule to interact with its substrate, the I kappa B, NF kappa B inhibitory complex, and cause it to fall apart in the cytoplasm to produce active NF kappa B. Now, what we have done is, see if I can change that, model some new molecules. Here you see it on the right here, that have a very interesting profile. And that when we look at our modeling now, we see that these molecules interact exclusively with the ubiquitin-like domain. And we know from work from May's group some years ago that if you carry out mutations within this binding area, this domain, it has a dramatic effect on the ability of the IKK beta to phosphorylate P65 in the NF kappa B complex, inhibitory complex. Two of these molecules, 7B and 7K, you can see they're different from the R group and the aromatic ring, interacted very specifically with the ULD site, no interaction with the KD site or the scaffoldimerization domain site, and no interaction with the NEMO amino acid sequence. So very specific for the ULD. And you can see the amino acid interactions here are very, very similar. We thought that these molecules probably would not affect phosphorylation, and we looked at this in M9 ENL cells and compared it with parthenolide. With parthenolide, you see a slight decrease in phosphorylation of P65 in M9 ENL AML cells. But when you look at 7B over this concentration range, it was a concentration range of 2, 5, and 10 micromolar here, you see no change at all. So this seemed to jive. What we couldn't figure out was why it was so good in the NCI human cancer cell line screen. It was able to inhibit the growth, very potent growth inhibition of about 91% of all the 60 human cancer cell lines in the NCI 60 panel. That was a very interesting molecule. So we thought, well, maybe it's acting at the ULD site, but it is affecting downstream the ability of NF-kappa B to be activated in the cytoplasm. And we thought that the only way to find out would be to do the DNA binding experiment and determine whether this particular molecule or these molecules these two were able to inhibit the binding of NF-kappa B to DNA, even though it looks like it's not affecting the inhibition, it's not causing inhibition of the phosphorylation of P65, which would block the ability of NF-kappa B to be activated in the cytoplasm. And sure enough, we found that there was a decrease in the DNA binding of NF-kappa B to DNA in the presence of this is molecule 7B, the one with the methoxy group in the para position. This was a more potent compound of the two. And so this molecule is actually affecting the NF-kappa B pathway and inhibiting activation of NF-kappa B, but it's not inhibiting the phosphorylation of P65. And so we assume that there must be some way in which, I can't go to the next slide. Well, I don't know. It's not. There we go. Okay. So there must be some way that this molecule is interacting with the NF-kappa B pathway that is unique. Initially, we thought that the ULD site might be a site that would be an allosteric site and would allosterically inhibit phosphorylation of the NF-kappa B complex, but obviously that was not the case because we do not see any inhibition of phosphorylation. But May's work indicates that when you look at the NF-kappa B pathway, what was my pointer gone? Here we are. Hard to see there. When you look at the NF-kappa B pathway, and this is the phosphorylation of the inhibitory complex. There's the I-kappa B with P65 and P50. It's a substrate for the kinase complex, IKK beta, IKK alpha, and NEMO. These are the 4 subunits of the kinase. So normally you would see an interaction of I-kappa B inhibitory complex, which is here, with this kinase, and then there would be a double phosphorylation, one on P65 and one on I-kappa B, and then this would lead to the ubiquitination of the inhibitory complex. But in May's work, it was clear that at this particular point, the dissociation of the kinase complex does not occur. It's still attached to the inhibitory complex, even when the ubiquitination occurs. And then the next step, once the ubiquitination occurs, is that the proteasome, the 26S proteasome, would recognize that ubiquitination tail on the I-kappa B inhibitory complex and would bind to that, but you would still have all these elements combined together. But once the proteasome bound, then the complex would dissociate and I kappa B would be degraded by the proteasome, and active enzyme would be regenerated, as well as NF kappa B to bind to DNA. So there's a lot of activity going on here because all these units stay together until the very last step when the proteasome interacts with the ubiquitin tail, and then you get a dissociation of all these individual units. What we think is that this compound 7H from the previous slide, this molecule which does not inhibit phosphorylation, is likely inhibiting one of these events that you see with the blue arrows, 1 to 5, and they're listed down here. Since we do inhibit NF kappa B binding to DNA, this last step here, then what probably will be happening is that the molecule is interacting by one of these 5, I should say 4 different ways. We know that it's not inhibiting phosphorylation. So in step 1, that's still occurring because there's no effect of 7B on that process. We've already shown that from the Western blot. But it could be that it's blocking the ubiquitination of that complex once the phosphorylation has occurred. And that could be the reason why you're blocking this whole pathway. Or it could be that it's blocking the interaction of the 26S proteasome. That's the next step, step 3. Or step 4, it's blocking the dissociation of all the units within that complex that has the proteasome and the inhibitory complex and the kinase. Or lastly, it's preventing the NF kappa B, the active NF kappa B now, from interacting with DNA itself. So we got to unravel all this, and we're hoping that we can make some sense. But the interesting thing is that the 3-dimensional modeling that we used in the IKK protein structures led us to this stage where we could actually manufacture a molecule that was very specific in the interfaces that it interacted with on the IKK monomeric molecule. So we conclude from this study, it is complicated and the targets are very complicated, but we did focus on determining the mechanism of inhibition of these sesquiterpene lactones to improve inhibition of the NF kappa B pathway when compared to parthenolide as anti-cancer agents, and we have achieved that goal. But what we know now is that one can actually design molecules to bind to specific interfaces on the IKK beta kinase complex to act as inhibitors of IKK beta NF kappa B activation in different ways, ways that don't involve the inhibition of the actual phosphorylation event that is mediated via the IKK complex. So I think this is a really good example of how molecular modeling can lead us to the stage where we can design interface-specific molecules to interact with proteins that are very complicated and have multiple binding sites and lead to the design of inhibitors that will inhibit the interaction of this very important NF kappa B molecule to the DNA apoptotic site. Okay, that's any questions?
Thanks. Thank you, Dr. Beecher, for sharing your presentation, knowledge, and experience. And thanks for highlighting the very useful key points for your research. and it's very interested, very rich area to the point. Actually, I lost the track about the time. So for time consideration, we will allow about 2 questions. And I do apologize from the audience. So if you have a question, please drop your question in the chat box or raise your hand to ask your question directly.
If you don't mind, Dr. Samra.
Yes, please go for it.
Thank you so much, Dr. Peter. Thank you so much, Dr. Raed, for a very nice, interesting presentation. I want to ask Dr. Peter a question. Possibly you might consider it as a naive question, but really I really astonished by this magnificent work that you have just displayed for us. So we have some of the students, MSc students, who are working on medicinal plants. So I want to ask you, how do you start thinking about such horrible work? Do you start selecting the medicinal plant or the active constituent, or how do you start planning such project? Do you count on the traditional uses of the medicinal plant, or you select the active constituent of the plant and then you build up your hypothesis on?
Well, it's a little bit of both, actually. I think once we latch on to a molecule that we know a lot about in terms of mechanism, and we know that the mechanism is unique, but the molecule has some problems, then what we try to do is either search the plant for molecules that are better drugs, that is, that they're more drug-like, and usually that's pretty hard to do when you're talking about sesquiterpene lactones, because most of those lactones, those sesquiterpene lactones are very, very water insoluble, and they're active in cell culture experiments because you can actually dissolve them in solvents such as DMSO, and so there's no issue with getting them into the cell. But what we found is that the best way is to try to improve on nature and take these molecules and modify them slightly so that they still retain their pharmacological activity, but they become better drugs. And this is the example of DMAP, that we kept the properties of parthenolide in the DMAP molecule and found that it was a much better drug because it had great water solubility and it even had low toxicity, just like parthenolide. And we were able to get it into a phase 1 study, clinical study. We're hoping that we can do the same thing with these modifications that we're carrying out now, and that was the effort when we tried to make these molecules. We then went on to introduce an amino moiety into them to see whether we could maintain that activity that we'd designed into the molecule, but improve the drug-like characteristics of the molecule so that we would have a drug that was able to withstand the rigors of the drug development process.
So you do
I'm sorry?
Expect that the active constituents and the active constituent in the 3 plants, Magnolia, Tansitum, and the other one, they have possibly the same pathway, the synergistic effect on glutathione pathway and the IKB?
Yes, they seem to have a similar mode of action, and I think the reason for that is that it's due to that very unique lactone ring system that is present in all 3 molecules.
The amine moiety.
Exocyclic double bond is a very electron deficient moiety, and that carbon, the terminal carbon of that double bond is able to interact with nucleophiles. And if you remember the modeling of the glutathione enzyme, GCL, I showed you from the Schrödinger suite, the modeling that showed there was an actual covalent bond formed when parthenolide interacts with the modulatory protein, preventing that formation of the disulfide bond. That's because that double bond actually interacts with cysteine and forms a carbon-sulfur bond that covalently binds parthenolide to that cysteine residue and prevents it from interacting with the other cysteine residue on the catalytic moiety. So I think that's the reason why these molecules work so well, is that they all have that same ability to interact with nucleophiles because of the electron deficiency of that lactone moiety with the exocyclic double bond. That's the active part of these molecules.
Okay, and if you are going to design another new molecule which is similar to this parthenolide, do you prefer to endorse the inhibitory effect on the IKKB mechanism or the glutathione pathway?
Well, we check both of those things because we know that even when you make these modifications and you make the synthetic analogs, these properties don't go away. All the molecules that I mentioned with respect to the NF-kappa B pathway, these same molecules also interact with the glutathione pathway. I just didn't have time to show you the data. But if you look at the publications, you'll see that whenever we publish on these molecules, we look at both the glutathione pathway and we look at the NF-kappa B pathway, and we usually see activity in both pathways simply because we still have that exocyclic double bond in the lactone. That's responsible for both the NF-kappa B activity and the glutathione inhibition. So the simple answer is yes, we can maintain that dual activity, and we've got evidence to show that. And it's good because that's synergistic and it gives that molecule that the anti-cancer characteristics that are unique to these sesquiterpene molecules.
Yeah, it is really interesting. Thank you. Thank you so much.
Thanks, Dr. Inham. Thanks, Dr. Peter, for answering all the questions. And for time consideration, we are not taking any further questions. And I will handle the session to my colleague, Dr. Etmar and Dr. Ala. So go for it. Thank you.
Thank you, Dr. Wathiq, Dr. Samra, and welcome everybody. We're going to start our oral presentation session. However, I want to grab your attention that tomorrow the conference is going to start at 5, Baghdad timing. And also there will be an electronic voting for the best oral presentation of the students and lecturers within the oral session, within the oral presentation session. We're going to start our oral presentation with Qsr. Qsr Muthanna Ahmed works at the Department of Pharmacy at Rashid University. Qsr is going to talk about molecular docking and synthesis of some new 5-member ring heterocyclic compounds with in vitro antiproliferative evaluation. Qsr, are you with us? If you are with us, you can start your presentation now, and you have 10 minutes only. Can you hear me, Qsr? We cannot hear you. I think you need to unmute. Okay. Your voice is a bit unclear. We still are unable to hear you properly.
Sorry, but I believe that Qsr needs to log out from Zoom and go back again and join with the audio to be able to make us listen to her voice.
Okay. While we're waiting for Qsr, I just also want to remind you again that tomorrow we're going to start at 5. Today, we're going to have 4 oral presentations, and at the end of them, we're going to have a discussion. So during the presentations, you are free to write your questions on the Q&A or on the chat, whatever you like. And tomorrow, we're also going to have another 5 oral presentations. And at the end of them, we're going to compare between the 10, and we're going to do an electronic voting to choose which one is the best, although all the research done and all the presenters are good. So we are waiting for. Okay, Qsr?
Okay, the sound is okay?
Yes, it's okay. You can start sharing your slides now. Yes, and you have 10 minutes only.
The screen is clear?
Yes, it's clear.
Thanks. Let me start with the project that's supervised by the Professor Dr. Munder Faisal Mahdi and Assistant Professor Dr. Ayad Mohammed. Here are the outlines of the project, introduction and aim and strategies, then results and discussion, and finally the conclusion. As we all know, cancer is a condition characterized by abnormal cell proliferation, and cancer remains the most complicated illness and the main cause of mortality in the world. Breast cancer is the most common cancer in women. The risk factors include family history, unhealthy lifestyle, old age, and obesity, and other risk factors. The breast cancer can occur due to disturbances. Estrogen disturbance is called ER-positive breast cancer, and can also be caused by progesterone disturbance, and the cancer is called PR-positive breast cancer. And if the cancer is caused by a protein called HER2, which means the human epidermal growth factor receptor 2, it will be called HER2 positive breast cancer. Here are the therapeutic options, which includes surgery, chemotherapy, hormone therapy, HER2 drug targets, and radiation. The selection of the therapeutic option depends on the stage and the type of breast cancer. Why do we choose pyrazole as a scaffold in our work? Pyrazole, as we know, is a 5-membered ring heterocyclic compound with 2 adjacent nitrogen atoms. Pyrazole is an aromatic system due to the delocalization of the 6 electrons inside the ring. Thus, it's stable. And this property makes it a good nucleophile, which makes it readily bind with the variety enzymes and receptors inside the body. The aim of the work, first step, is to synthesize a new series of chalcone, pyrazoline, isoxazoline derivatives from the attractive drug naprometon. The second step in the work includes docking studies that were performed by Gold Suite software on ER-alpha protein. The third step is the estimation of ADME, absorption, distribution, metabolism, and elimination, using a Swiss ADME server. And the final step is the evaluation of anti-proliferative activity against MCF7 breast cancer cell line. Here is the scheme of our work, which includes naprometon reaction with glycolic acid in the presence of ethanol and concentrated sulfuric acid to produce compound 1, which is chalcone derivative. The second step includes the reaction of chalcone with many different hydrazine derivatives to produce the isoxazoline and pyrazoline derivatives. And the third step is amidation reaction, which converts the carboxylic acid moiety to amide functional group. Results and discussion, molecular docking results. Here we see the binding energies of nabiomytone derivatives and the tamoxifen standard drug docked with ER alpha protein. As we see that 1E compound gives the highest PLP fitness score, 75.61, with arginine 394. Here is a photo from the Gold Suit software, which appears 1E compound, which gives the best result in cell line. And as we see, there's 2 H bonds with arginine 394. ADME study results. As we see in this table, nearly all the compounds are passively and highly absorbed from the GIT route of administration when taken orally, but except 1E and 2E compounds would have low absorption and can be given by another route of administration. And as we see also that all the compounds satisfy Lipinski rule. FTIR results of the 1E compound appears that the compound have 2 characteristic peaks. The first is appeared at 1672, which belongs to the C double bond N, and the second is appears at 1732, which belongs to the carboxylic acid carbonyl group. And here are the proton NMR results of 1E compound. Here we have 2 characteristic peaks belongs to B and C, including in the pyrazole ring, which appears at doublet at 2.58 and 2.74 chemical shift. And here we have the carbon NMR results, which appear that compound 1E have an important peak of C double bond N, which appears at 180. The in vitro cytotoxicity assay, MTT assay, is done against MCF7 human breast cancer cell line, which appears that 1E compound gives the best result and the lowest IC50 value, which gives 19 micromolar value, compared with tamoxifen, which gives 18 micromolar IC50 value. Estimation of percentage of breast cancer cell line death using tamoxifen as standard drugs in comparison with the tested compounds. Here we see that also compound 1E gives the highest cell death percent, 89.13%, and the compound 2E also give a good percent, 85.10%, and finally compound 2C also give 68.10%. Those compounds are compared to standard drug tamoxifen that gives 96.61%. Finally, the conclusion, novel compounds were synthesized and conferred by spectroscopic analysis. Evaluation of anti-proliferative activity against MCF7 was done, and compound 1E, as we said, is a novel compound and gives the most potent activity against breast cancer cell line. ADME studies showed that nearly all the compounds are good, are highly absorbed from the GIT, and all the compounds satisfy Lipinski rule. And the docking studies showed appropriate correlation with the in vitro results and compatible. And the compounds 1E and 2E was compatible with the docking results. Here are the references of our project. Thank you for your listening.
Thank you very much, Qsr, for this nice presentation, and thank you for sticking on time and being on time. The next presenter we have is Hani. Hani Aqil al-Husseini works at the Department of Clinical Pharmacy, Al Karama Hospital in Baghdad. Hani is going to talk about in silico identification of androgen receptor inhibitory drugs for the treatment of prostate cancer. Hani, are you with us?
Yes, Doctor.
Welcome, Hani. And you can start sharing your slides, and you have 10 minutes only.
Thank you very much. Hello there, I'm Hani Akil, PhD candidate, clinical pharmacology, College of Medicine, University of Baghdad. This presentation about in silico identification of estrogen receptor inhibitor drugs for treatment of prostate cancer. As we know, the prostate cancer, one of the major type of cancer that most patients suffering in Iraq and all over the world. Mainly prostate cancer is the cancer that occur in the prostate and the male, mainly over the age of 15. In this presentation, I'll talk about how to treat, and I'll talk about the binding affinity of prostate cancer treatment drug medication with the androgenic receptor. How this medication work, how this medication bind to androgenic receptor. Mainly, as we know, the prostate cancer treated by either surgical treatment or by hormone that blocking the androgenic receptor completely, that we call as androgenic blockers. Molecular docking study. In this presentation, I will talk about the molecular docking study. The in silico means molecular docking study. That molecular docking study of the bioactive peptide or chemical drugs molecular exerting their active by studying the interaction and binding with the receptor to provide the evidence and binding of the binding conformation. Recently, most of study are conducted on molecular docking study, and there are a lot of programs that have been done on the molecular docking study. Molecular docking is recently have been done by a very efficient program and very reliable and very close information and close contact with the new or with the real affinity and real binding. Here I'm talking about the benefit, why we use androgenic receptor antagonist for treatment of prostate cancer. Actually, prostate cancer is improved, not improved, but stimulated by androgen and by testosterone. For that reason, the first idea is how to prepare a drug that block the androgenic receptor. After blocking the androgenic receptor, actually the prostate cancer is attenuated. However, in this presentation, I'll talk about several types of androgenic blocker drugs, such as glutamide, nilotinide, and other. But most patients have resistant to these drugs. The cancer type is resistant to androgenic blocker. So how to overcome this resistance, or can we have an effective androgenic blocker drugs that bind effectively with the receptor and blocking the androgenic receptor? After blocking androgenic receptor, the cancer is attenuated and the body can overcome easily the cancer cell. So if they're resistant to treatment, this is called castration-resistant prostate cancer. There is no rule of other treatment. Sometimes the doctor thinking about castration. So what's the best drug for this for resistant case? Is there any drug for resistant case? For understanding how to treat the resistant case and the cases, and how to understand how to treat the resistance case, it is obvious that we have to know the binding of androgenic receptor itself with the androgenic blocker. So in this research, we have I have done the two types of androgenic receptor. First, the wild type androgenic receptor, which is the common in all over the world, and the resistant androgenic blocker, or as we say in the second point, that resistant to treatment, the type of androgenic receptor that resists to treatment by the androgenic blocker. So the aim of this study is to identification of androgenic receptor binding site, and using computational technique by some drug which is available for free to do molecular docking study and study the affinity of the drug. The third point is to compare the binding site between the mutated and wild type that are commonly. Recently, our recent or final aim is to respect what's the best androgenic receptor blocker that have higher affinity to block the androgenic receptor, which is may benefit over others. Material method, it is in silico experiment, has been done by a program Autodoc Vina, which is available for free. Autodoc tools also available for free. Bimol also have a free license available. Comparative from the Cambridge University, and Chimera and PYRX programs. These all programs has been collaborate to done this this research. However, protein structure has been provided from protein data bank, and also selection of pharmacological molecular that with the inhibitory potential to the normal and mutated receptor. So we will find out about what the potent drug, what the potent medication that block the androgenic receptor very effectively, even in the mutated and wild type androgenic receptor. Here we'll talk about a procedure. First, I collect the 3D structure from the of the wild type androgenic receptor, which is called T8778, from the protein data bank, and the wild type that may present in Iraq with the mutated one that may occur also in Iraq. Here is the website for the 3D structure protein, if someone wants. After that, we prepare this protein by using Autodoc Vina. This program is available for free. Autodoc tools is preparing the protein by adjusting the guide and select the resolution and also removal of water and adding polyhydrogen, adding Kalman charge, and also run this Autodoc Vina and interpretation of this result. Actually, most of research that I have seen in the field of medicinal chemistry, they use glide or gold because it is effective, reliable result, easy interface. This program Autodoc Vina is available for free, but its interface is very difficult. Okay, okay, the docking result. After docking with the wild type androgenic receptor by using Autodoc Vina, I first test the androgenic agonist drug with the receptor. Then find that the testosterone have the affinity of -10, and dihydrotestosterone also have high affinity for the androgenic receptor, for androgenic agonist with the androgenic receptor. However, what about the androgenic blocker drug, the binding affinity of androgenic blocker with the receptor? This is the blocking of androgenic blocker drugs with the androgenic receptor. Here, as we see, the affinity is high, except enzalutamide and ARN, which is apalatamide, which have low affinity. This result is making question in my head about, okay, why the second generation androgenic blocker enzalutamide and ARN 409 is low affinity for the normal androgenic receptor. After understanding why this normal affinity,
sorry, honey, you have 1 minute left. If you can speak up.
I will fast. I will end it fast. Okay, this is the binding affinity of second generation androgenic blocker, but at the other side of the receptor. As we see here, the docking result is done on the other side. This is the normal side of the binding of testosterone and dihydrotestosterone, and this is the other side that the second generation androgenic blocker have bind. The docking result of mutated show also that the nilotamide have high affinity for blocking the androgenic receptor, especially in Iraqi patients. The second generation androgenic blocker have high affinity also, but on the other side, the same region, the other side, other than the common site of the androgenic receptor. A docking result for a mutated protein that show that bicalutamide is not effective well in this patient with the mutated androgenic receptor, so it may not effective in treatment of castration-resistant prostate cancer. But however, flutamide and nilotamide is more effective for treatment, even than more effective than second generation anti-androgenic for treatment of prostate cancer, especially in Iraqi patient. Conclusion: we in this research understand the binding of androgenic receptor, understand that second generation bind at the other side, other than the common site of androgenic receptor, understand the most drug that may help us to treat the castration-resistant prostate cancer. Finally, I thank you. Thanks to Dr. Munder. Thanks to Mustansiriyah College of Pharmacy. Thanks the audience. If there are any questions, I welcome.
Thank you very much, Hani, for this nice presentations. And please, if you have any question for Hani, you can type it either on the chat or on Q&A. Next, we're going to move to Rand. Rand Abdel Karim works at the Department of Pharmacology and Toxicology at Al Rashid University. Rand is going to talk about comparative study for citrus fruits phytochemical screenings and in vitro antioxidant activity. Welcome, Rand. Are you with us? Can you hear me, Rand? Can you hear me?
Yes, I can hear you.
Okay, we can hear you as well. Just please raise your voice and you can start sharing your slides. You have 10 minutes, Rand.
Is the video on?
No, we still haven't seen your slides yet.
Okay. Is it clear?
I think it's, yeah, it's coming up now.
Okay.
Okay, you can start now, Ryan.
Okay, thank you very much. Good evening, everyone. And I'd like to thank Professor Dr. Munder Faisal and Assistant Professor Dr. Inam Sameh for giving us an opportunity to share our research, me and Professor Dr. Ihab Khalifa, that we both works in Al Rashid College
Department of Pharmacy. So now I will show the study which entitled the Comparative Study for Citrus Fruits Phytochemical Screening and In Vitro Antioxidant Activity. First, I will talk about the abstract. Citrus is well known for its nutrition and health promotion values. In recent years, the antioxidant activity of citrus fruits and their roles in the prevention of various human chronic and degenerative diseases have attracted more and more attention. The citrus peels are suggested the main source of dietary antioxidant. To have a better understanding the mechanism of the antioxidant in the citrus peel, we review a study of this antioxidant by measuring the phytochemicals and also introduce methods for antioxidant evaluation for the citrus peels. First, I will talk about the oxidative stress, which reflect an imbalance between the systemic manifestation of reactive oxygen species and biological systems' ability to readily detoxify the reactive intermediates or to repair the resulting damage.
Rand, can you make your slides full screen? They are not full screen to us.
It's a full screen. Sorry.
Maybe it's just me. Sorry. Okay, continue.
Okay, is it now okay?
Zoom it a little bit more. Zoom it and hide the toolbar, please.
Okay.
It's okay now. It's okay now. Yeah.
Okay. Sorry.
Yeah.
Okay. Antioxidants are chemicals that interact with and neutralize free radicals, thus preventing them from causing damage. Okay. In this slide, I show the causes or the main causes that will cause this oxidative stress. It may be from biochemical reaction, byproduct, or exposure to environment, and higher levels of dietary xenobiotics. All those causes will result in generation of ROS, or reactive oxygen species, and reactive nitrogen species, and this will cause oxidative stress in different pathophysiologic conditions. The phytochemicals that are present in plant extracts have potential antioxidant properties, which may be useful in therapeutic application. The citrus fruit is rich in antioxidants such as beta-carotene, ascorbic acid, terpenoids, alkaloids, beta-cytosterol, and polyphenols like flavonoids. The objective of this study is, it's indeed we make two extract of citrus fruit. The part we use the peel part of the plant. We have different, the same genus but different species. We have orange and pomelo. Orange is the botanical name, which is Citrus sinensis, and the pomelo Citrus maxima. Then from these two extracts, we determine the antioxidant activity by different methods. The phytochemical constituents of citrus, flavonoid, alkaloids, and terpenoids. The methodology. First, we prepare the citrus peel extract, the ethanolic citrus from both pomelo and sweet orange peel extract prepared by maceration method. By macerated 10 grams of the dry powdered citrus peel and 100 ml of 80% ethanol. After that, all extracted will be kept on cool for further investigation. Also, we do phytochemical analysis, which is a preliminary screening, chemical test for flavonoid, drag and drop for alkaloids, and chemical test for terpenes. In relation to the antioxidant activity, we do total phenolic estimation by using the Fullen CO-Cal2 method, and also total flavonoid content. The estimation of the flavonoid was carried out by aluminum chloride colorimetric method. The blank and calibration curve was obtained by using 80% alcoholic solution that contain flavonoid. We use quercetin as the flavonoid that will be the standard solution to determine the flavonoid content of the sample extract. Also, we use a simple and sensitive, it's really very popular method for determination of antioxidant activity, which is by measuring by using DPPH assay, diphenylbutyryl hydrazine assay. We take both citrus peel extract, pomelo, and the orange extract, and various concentration of these two extracts were prepared by serial dilution. Then the ethanolic extract will be stock solution for 4,000 microgram per mol. Ascorbic acid, vitamin C used for comparison or as positive control. The DPPH solution in the absence of the extract used as the control, then measure the absorbance in the UV spectrophotometer. The result and discussion. First, we have the result of the preliminary phytochemical screening. We have positive test for both extract, pomelo, and orange for alkaloids, flavonoids, and terpenes. Then the result of the quantitative determination of the total phenolic and total flavonoids content for both citrus peel extract. The pomelo peel extract exhibit or show higher total phenolic content, 74 milligram per gram, while the orange peel extract show lower total phenolic percent by 45 milligram per gram. While the total flavonoid content result show that the pomelo peel extract was 21 quercetin, milligram per gram quercetin, compared to the orange peel extract that will be 23 milligram per gram quercetin. The qualitative DPPH radical scavenger activity, the results show that pomelo peel show higher antioxidant activity compared to orange peel. Moreover, citrus peel possessed higher antioxidant properties, except a significant dose-dependent inhibition of DPPH radical, and the solution color change from the dark purple, to the light yellow. In this slide, we show the dose-dependent antioxidant activity for both extract and the standard that we use, which is vitamin C in the green color, orange in the blue color, and pomelo in red or light red color. Also here, we determine the concentration of the extract that require to inhibit 50% of the initial DPPH free radical, the IC50 for pomelo was calculated 287 microgram per mol, while for orange peel extract was 341 microgram per mol. This slide shows the IC50 value of vitamin C, which was 260 microgram per mol. Vitamin C we use as a standard antioxidant. As we know, the higher DPPH radical scavenging activity is associated with a lower IC50 value. Previous studies show that sample which had IC50 lower than 50 microgram per mol was very strong antioxidant. Between 50 and 100 microgram per mol was a strong antioxidant, while between 100 to 100.5 microgram per mol was medium antioxidant, whereas the weak one below than moreover than 150 microgram per mol. The conclusion of this study, we conclude that the pomelo peel show higher antioxidant activity compared to orange. Citrus peel possessed high antioxidant properties, and it's potentially rich source of natural antioxidants in scavenging DPPH and reactive oxygen radicals. Thank you for your time.
Thank you very much, Rand, for the nice presentations. And if you have any questions for Rand, please type them at the Q&A and we can answer them at the end. Now I'm going to transfer the mic to my colleague, Dr. Ola, to finish with the next two presentations. Dr. Ola, the mic is yours.
Thank you. Thank you, Dr. Asmar. Good morning or good afternoon, everybody, depending on where you live. The next presentation is on organization factor influencing the nurse ability to prevent and detect adverse drug events in a public hospital in Iraq, and will be delivered by Dr. Ali Aziz, who holds PhD in clinical pharmacy and is one of the leading teaching staff at the College of Pharmacy, Baghdad University. Thank you. Dr. Ali, can you hear me, please?
Thank you so much, Doctor. Thank you so much for the University of Mustansiriyah College of Pharmacy for inviting me to present my study. So let's start with the So today we'll talk about organization factor influencing nurse ability to prevent, detect adverse drug events in public hospital using patient safety model. So this study already published in the Saudi Pharmaceutical Journal. I would like to acknowledge my co-authors, Dr. Sara Aboud from the University of Kufa, Ashwaq Tajat from University of Sulaymaniyah, and pharmacist Hind Muwaffaq, pharmacist Fatima Rahim, and pharmacist Zahra Ali. So System Engineering Initiative for Patient History, it was developed in 2006 by Karian and et al. And it's really helpful to identify factors influencing the outcome. So we are not talking about only individual factors. We're talking about the organization factors influencing the outcome. Because we usually, when we have bad outcome, we blame the person. But here we need to understand the whole story about the factors influencing the outcome. I used this model also at the University of Iowa in my PhD study in 2017 to investigate the factor influencing adverse drug events in nursing homes. I also use it in this study to identify the factor influencing nurse ability to detect and prevent adverse drug events in Iraqi hospital. So the objectives is really use this model, which is called System Engineering Initiative for Patient Safety, to understand the factors influencing the nurse ability to detect and prevent adverse drug events in our public hospitals. So the method was cross-sectional study. The study was multi-centers. We used we distribute the paper survey to 11 hospitals, 9 of them in Baghdad and 2 of them in Babylon. So it was self-administered. This happened in October to December 2019. We created our survey, relied on this system, which include factors related to person, organization, tools, task, and environment. So as I mentioned, 9 hospitals in Baghdad and 2 hospitals in Babil provinces. So why we need to understand all the factors? Because when we have problem or we need to identify the shortage or the limitations, we need to understand all the factors will influence the adverse drug events. So the factor could be personal, related to the, when I say personal, related to the nurse ability to detect adverse drug events, their education, their skills, their training. Also, when we talk about the task, we talk about the time available for this task, the workload. About the technology and tools, we need to understand the available tools for monitoring, blood pressure meter, blood glucose meter. When we talk about the organization, we talk about the collaboration among healthcare providers. when we talk about the environment in the hospital, as you know, we have the noise, we have interruption. All these may influence the nurse ability to detect and prevent adverse drug events in their hospitals. So our sample was like more than 600 nurses, majority were male. And as I mentioned, majority also in the hospital in Baghdad. Most participants, they had diploma or nursing schools. And they have title of skilled nurse, technical nurse. We have a few university nurses. Also, the majority were related to the internal wards specialty, and most of them, they work for the day shift. Our research team, they went also during the night team, but they collected less than the morning team, shift. So from this table, you can see the nurses were able to identify serious adverse drug events in the last month. So it's required to report adverse drug events in their hospitals. Some of them, they were fearful. They were fearful from reporting because, you know, we live in a blame culture, and they were afraid to report the adverse drug events and medication errors because they may get blamed about this is your fault and you did it. That's why they avoid reporting. So as you can see, when the adverse drug events happen, they usually inform the physician more than informing pharmacists or treated, because, you know, everything should be prescribed or ordered by physician. And mostly they measure the blood pressure twice a day within the hospital. I'm talking about inpatients, not outpatient. So usually they measure the blood pressure. Why I care about blood pressure? Because more monitoring, more expecting, we can avoid adverse drug events. We can detect any hypotension. We can detect any hypertension. So we need to increase the frequency of monitoring. Here, it's related to the personal factor. As I said, we have 5 factors, 5 domains of this model. Any of them could influence our outcome. So I'm talking about each one. The first one is personal characteristics. As you can see here, less than 50% of them, they have less than 50% of them, they have experience in monitoring, in detection adverse drug events. Also, less than half, they receive training. They have not enough experience. They haven't received training, but they can measure the vital signs. But in general, not enough training. Also, they haven't received training. They have also, as you can see, less experience in detecting. So the next slide talking about the task factor, the second factor. The task factor, as you can see in the first bar, most of them, they say we have lots of work. They are overwhelmed. So how can we expect they can detect or prevent if they are overwhelmed? Also, majority of them agree they need to work fast. And finally, they say, some of them, we have enough time, but they work fast. They are overwhelmed. So imagine with me the situation in the hospital. The next factor will be the tool. Do we have enough tool to detect or monitor patients? As you can see, we care about 2 meters, blood pressure meters, blood glucose meters, and both of them, less than 40%, they say we have adequate number. So in general, we don't have adequate number of blood pressure meters, blood glucose meters. Imagine the situation. This is the third factor. The fourth factor, organization factors. The collaboration among healthcare providers are okay, but we have one problem here. We don't have adequate number of nurses per patient. So we have not adequate ratio of nurse to patient. So we don't have enough number. Imagine with me now, we will have clear picture about the organization factor. The last factor, which is the fifth factor, the environmental factors. As you can see here in the first part, they have interruptions. Half of them, they say we have interruptions during the administration of medication. Someone will talk with you. You will make medication errors. Also, they have distracting noise. So as you can see with me, the 5 factors could influence the outcome. We measured the factors associated with the adverse drug events using binary logistic regression, and we have 2 significant factors. One of them is adequate nurse experience to detect adverse drug events will reduce the incidence of adverse drug events. So we have negative association. Nurse-physician strong collaboration also reduce the incidence of adverse drug event. So also we have negative significant association. So in conclusion, we have shortage in nurses. I'm talking about public hospital. Inadequate nurse experience in adverse drug events detection, no training to not enough experience, they haven't received training. I'm talking about adverse drug events detection, not about something else. Fear of reporting. So we have underreporting because they are afraid from blaming. Finally, we have shortage in monitoring equipment such as blood pressure meter or blood glucose meter. We have 2 significant factors could influence the incidence of adverse drug events. Good nurse-physician collaboration will reduce the adverse drug events. Longer experience of nurse in adverse drug events detection could help to reduce the adverse drug events incidence. So our recommendations, we have 3 recommendations. We need to increase the ratio of nurse to patient. We don't need nurses to be overwhelmed. Secondly, provide more monitoring equipment, blood pressure meter, blood pressure, glucose, all the lab data required. Finally, providing training course to nurses. This is our role as pharmacists in hospital. Please train your nurses at your hospital about how can we identify adverse drug events such as bleeding, hypotension, any physical or psychological adverse events happen by any medication, so they can detect it and report it so we can treat it to avoid any bad consequences. Thank you so much for Mustansiriyah University College of Pharmacy for having me, and happy to receive any questions in the chat later. Thank you so much.
Thank you so much, Doctor. I well rehearsed. I like your presentation and recommendation, and I do agree with the is on the effect of molecular imprinting on the loading and release of poorly water-soluble drug on hydrogel contact lenses, and will be delivered by Zahra Yahya Sabri, who is a master's student at the College of Pharmacy, Mustansiriyah University. Thank you. Zahra, can you hear me?
You hear me, Doctor? Hello?
Thank you. Excellent. Thank you.
Thank you very much. I am Zahra Yahya, MSc student in Mustansiriyah University. Sorry.
Yes, I can hear you. Go. Thank you.
My research is the effect of molecular imprinting on the loading and release of poorly water-soluble drug on hydrogel contact lenses, supervised by Dr. Athmar Daher. I'm going to talk about introduction, experimental work, and result and discussion conclusion. Treatment of ocular diseases is usually through eye drops. However, in the eye drops, only 5% of the instilled drop will penetrate the ocular tissue, and the other 95% will removed quickly. The reason for that is the poor corneal penetration. The cornea was a vascular tissue that covered the eye, made of several layers that prevent drug penetration. Loose through conjunctiva, which is the white part of the eye, excessive tearing and blanking, which allow the drop to get out of the eye, nasolacrimal drainage, and systemic circulation, which results in many side effects. So as a result, the residence time of eye drop in the contact with ocular tissue will be short, approximately from 2 to 5 minutes, due to these barriers. To improve the ocular bioavailability and residence time of a drug in ocular tissue, many research using thickening agent in situ gelling, multi-adhesive polymers, ocular inserts. And we're gonna focus on one type of ocular insert, which is a contact lenses. Contact lens may have many types, and the main type is their widely used in is a hydrogel contact lenses. There are polymeric cross-link hydrogel device, widely used for aesthetic and vision correction, could be used as drug delivery system for loading and release of a drug. This figure shows the difference in residence time between the eye drop and contact lenses. After 8 hours, the drug particle was completely washed in eye drop, while a large amount of drug remained in contact with ocular tissue in contact lenses. Advantage of contact lens is highly biocompatible, increased bioavailability up to 50%, reduced dose and dosage frequency. Also, systemic absorption and side effects are reduced, can be used as a reservoir for sustained release delivery of drugs. The disadvantages of contact lens or the problem that affect on drug loading if the drug is hydrophobic due to high water content of contact lenses, and the fast or burst release if the drug is hydrophilic. So there are many techniques to enhance loading of hydrophobic drug into contact lens. One of the most important methods is the molecular imprinting. As seen in this figure, the drug molecule was mixed with the monomers during contact lens preparation to make a cavity similar to drug structure. Then the drug template was removed. So once the contact lens was soaked in drug solution, the drug will incorporate it again into these cavities. The drug of choice for introducing into contact lens is itraconazole, which is beyond to class 2 by pharmaceutical classification system, have highly permeability and very low solubility. It is antifungal agent widely used to treat fungal keratitis, available as 1% eye drop, applied 1 to 2 drops every 2 hours, which required multiple dosing, so may lead to poor patient compliance. The aim of the study is to formulate itraconazole releasing contact lens to extend the release and enhance precorneal residence time. Molecular imprinting will be used to enhance the loading of hydrophobic itraconazole. Experimental work. Contact lens preparation, as seen in the table, different ratio of HEMA and MAA monomer was mixed. Then the cross-linker was added. For the molecular imprinting contact lenses, itraconazole was added. Finally, we added the initiator. This mixture was then injected into a mold and put it in oven for 6 hours. Then the obtained hydrogel foam was washed several times to remove the drug template and the unreacted monomers. Then the prepared contact lens was characterized for their optical transparency, folding endurance, swelling ratio, oxygen permeability. Also, the solubility of itraconazole in different solvent was measured to select the best solvent for loading the drug into the contact lens. The loading was done by soaking the dry lens into concentrated solution of a drug, leave it for 24 hours, then it was removed, raised and dry. For quantification of total amount of a drug loaded into the lens, methanol extraction was used. The release of a drug from contact lens was done by placing the loaded lens into 15 mol of phosphate buffer saline, pH 7.4 plus 1% sodium lauryl sulfate. The whole volume was replaced by fresh media at predetermined time interval for 24 hours. The results. Here is a picture of our prepared contact lens. The lens was soft, transparent, and free from bubbles. This is the same for all molecular imprinting and conventional contact lens. The folding endurance test was performed to measure the durability of contact lens. Opening and folding the contact lens until the contact was cracked or past the 250 times without a break. All the prepared contact lens will pass this test without a break. The oxygen permeability of contact lens is essential for the contact lenses to avoid hypoxia and related adverse effects, especially for long-term used contact lenses. Decay is indicated for oxygen permeability. High decay indicated better permeability. The red color in the table, F3 and F3MI, have the highest decay value. Also, molecular imprinting not effect on the oxygen permeability. As we see in the table, different solvents we used to measure the swelling ratio of prepared contact lens. Swelling ratio increase with increased MAA up to 8% F3, followed by reduction in swelling ratio because the lens was reached to the maximum stretching. Also, there's no difference in swelling ratio between the molecular imprinting contact lens and the conventional contact lens with the same composition. The maximum swelling ratio was seen in ethanol water, methanol water solvents. The loading of the drug depends on the solubility of the drug and the swelling ratio of contact lens in the loading solvent. So when we study the solubility of itraconazole, the highest solubility was observed in acetonitrile. And the swelling ratio of contact lens was relatively high in this solvent. From the table, the amount of loaded drug was enhanced by 3.7-fold by using molecular imprinting method. Also, F3MI contain 8% MAA have the highest drug loading. When we study the release of itraconazole from the conventional contact lens, around 40% was released within the first 2 hours, which considered as a burst release because of the small amount of drug loaded into these lenses, while the release of itraconazole from molecular imprinting contact lenses, the drug released within the first 2 hours was relatively reduced to 20%, and the release was extended between 40 to 80% within the first 24 hours, which allowing to wear the contact lens for more than 1 day. As a conclusion, itraconazole releasing contact lens was successfully formulated using molecular imprinting. Molecular imprinting does not affect on the property of the lens, such as the transparency and oxygen permeability. Molecular imprinting contact lenses has a 3.7-fold increase in itraconazole loading compared to conventional contact lenses. F3 molecular imprinting with 8% MAA was chosen as the optimum formula because it has the lowest burst release and only 40% of the drug was released within 24 hours. Thank you for listening.
Thank you, thank you. Now, if you have any question or comment, the researcher would be happy to answer them.
You can type your comments on the chat or on the Q&A, or you can raise your hand.
Okay.
I cannot see any questions on the chat or on the Q&A. So this is a question for Rand. Rand, are you still with us? Can you hear me, Rand? Are you still with us?
Okay.
Rand. If you can hear me.
Can you hear me, please?
Yeah, I can hear you now, Rand. I have a question from Dr. An'am. So, do you think that vitamin C was the best standard reference drug that can be used in your research?
In this method, we should use antioxidant, and the best popular non-antioxidant vitamin C and vitamin E because of its properties as an antioxidant. So I think it's vitamin C or E can be used as an antioxidant in this research.
Thank you, Rand, for answering.
Dr. Athmar, excuse me, can I ask a question for Rand as well?
Yes, please go ahead.
Yes, Doctor.
Rand, I want to ask you, I have only two questions. Why did you use the maceration technique for extraction?
Yes. Maceration technique is one of the extraction methods that we use in plant extraction, because it's a cold method that may be phytochemicals that will affected by heat also may cause a broken to some phytochemicals like flavonoid sensitive one. So we use macerated. It's a little bit not fashion method, but we can say it's safe extraction method.
That means you tested another techniques?
Yes, we can use chocolate extraction, which is the heat method, hot method, sorry. But we use the maceration in case we have a sensitive phytochemicals. And of course, we have hesperidine that's sensitive to heat as a flavonoid.
Great. Thank you very much. Thank you, Rand.
Thank you, Rand. We have a question or a remark from Dr. An'am to Dr. Ali saying that I think unbalanced text differences might affect the final conclusion. Are you thus, Dr. Ali? And do you agree with Dr. An'am?
Hello, Dr. Athman. Thank you, Dr. An'am. Unbalanced on what, Dr. Athman?
Unbalanced text difference might affect the final conclusion?
I don't think so, because we already tested the gender and there was no significant association in the final, in the logistic regression. The gender has no significant association with the outcome, because, you know, the majority may be the nurses, as we know, but when we visit them, the male were more cooperative with us. So it's a convenience sample. So convenience sample, you only get what you get, you know. So it's without incentive, without giving them any incentive. We have no enforcement on them to participate. So we only invited them, and anyone were comfortable with the questions, answered our questions. Some of them, they were maybe afraid from the blame culture. You know, in Iraqi culture, they usually,
I know.
they have, you know, kind of
it's very hard to get information from hospitals.
Especially if you are asking about adverse drug events, medication errors, because they don't want eventually to be blamed. Like they don't want to declare something happen in their ward or hospital. They get so get published with their names or something like that, or with their hospital. So we try to comfort them about this will be anonymous. We will not mention your hospital per se. We only mention the name of the hospital without specifying the number or percentage of adverse drug event per each hospital, because our role is not to blame or to identify which hospital has the highest percentage. Our goal is to give picture. We have more than one factors could influence the outcome. So we have, as I said, organization factors, environment factors, personal factors, task or workload factors, and technology or tool factors. So we usually blame person, but the person maybe will not, cannot work alone without good environment, good collaboration, available tools, enough number per patient. So I think this system is very helpful for any, I think, setting to say, if we need to improve the outcome, we need to look at the comprehensive pictures, not only at the person or at the employees.
At the whole picture.
The whole picture, because, you know, if you have deficiency or you have limitation in one picture, this will affect the whole story. That's why I think this is the whole story from my presentation, or to say, for any problem, we need to look at the system, not to blame the employee.
Thank you. Thank you very much, Dr. Ali. Dr. Ghazi, you are welcome to ask the question.
مساء الخير.
مساء النور.
Dr. Ali, thank you for your very interesting paper. Would you mind if I ask you a couple of questions, please? I may miss some of those things you have mentioned, but just bear with me and let me ask you the first question. Did you take into consideration the number of beds occupied between those hospitals? Because the error can be due to the number of beds occupied. When you compare two hospitals, one of them is like 100% or 90% bed occupied. The other one is like 70%. That's the first part of the question. The second part of the question is related to the same thing, which is, did you try to make, I mean, maybe you did, but I don't know if you find a significant difference between the error reported during night shift versus the day shift, because I heard that you have mixed those shifts, night shift nurses and day shift nurses. And I think there is sometimes differences between, that's biological differences, that error can be different from time to time. And thank you so much, Ali. Appreciate your help.
Thank you so much, Dr. Ghazi. Quickly, our goal was not to compare among the settings. We didn't measure the significant difference between the hospitals themselves because it's not our goal. Our goal was to identify the factors influencing, the comprehensive factors for all the hospitals influencing the outcome. So the goal was not to compare between the hospitals. We took for each participant, we took the outcome variables, and also we took the independent variables. When we conducted the logistic regressions, we tried to find out the relationship or association between those factors and our outcome, which is the number of reported adverse drug events within the last 30 days or in the last month. So the outcome was like it's self-assessed by the nurses themselves. They put the number. We didn't judge them. We didn't do retrospective chart extraction to find out the actual number. So the numbers were declared by the nurses by themselves. So it's not like retrospective chart extraction. We went and we reviewed, as I did in my PhD study. I went there, I reviewed the charts, I found out the real incidence of adverse events. But here we asked them about the adverse events they detected in the last 30 days. So I think because if we can include such analysis, but we will have, sometimes we have limitation number of tables for the publications. So journals, they don't like to have 10 tables because if I go on and on with the sub-analysis, I could have 10 tables eventually. So I try to focus on the couple tables of demographics, and also our goal was to identify the factors associated with the adverse event. So if my goal was to identify the amount hospital, I could do more analysis. But for sake of limitation and number of tables, I stuck with those tables, and unfortunately I didn't do those analysis. Thank you, Dr. Ghazi, for your question.
Dr. Ola, would you mind if I just go and just ask one more question? It's just for the interest of Dr. Ali. Dr. Ali, would you plan to extend this paper or this research for the future?
We already did, Dr. Ghazi. Thank you. We already did with the pharmacist, and it's in
very good. I was going to ask you the same question. I was going to ask you the same question. It's very interesting. Ali, I congratulate you. You are a very smart researcher. Thank you so much. Thank you, Dr. Ola.
Thank you so much.
Thank you. Thank you, Dr. Ghazi and Dr. Ali. We have a couple of questions for Zahra. Zahra, are you still with us?
Yes, Doctor.
The first from Dr. Widad. She's asking, what about the solubility of the drug in contact lens? Did you have result about it?
The stability, doctor. The stability of the
stability, sorry.
Yeah,
sorry. The stability of the drug in contact lens. Did you have result about it?
What did you mean for a long time?
Yes, for a long time. Like, is the drug stable in the contact lens for a long time? Have you done that or not yet?
Not yet.
The other question is, how about eye irritation? Dr. Mohammed is asking, you are using an organic solvent, so it may cause eye irritation and sensitivity. What do you think? Do you think it's going to cause eye irritation and sensitivity?
I will wash the lens after soak in organic solvent several times to remove the solvent. I use an loading solution, and the loading solution will remove by washing several times. Then I put in phosphate buffer saline for the drug study. So there's no, the solvent will evaporate it completely.
So the solvent is evaporated and removed completely. Thank you very much, Zahra. I don't know if we have any other questions. I cannot see any questions in the Q&A. If you can see Dr. Ola on the chat, is there any question? If not, so we can end up the day.
Yeah, I just have one final comment for Dr. Ali Aziz. I like the idea of the research. It shed light on a very important clinical problem that we have in our hospital, which is shortage of staff and limited resources that we have, as he highlighted. So I think prioritizing the patient need is a key factor. So we can prioritize patients. So these patients, their condition, their clinical condition is critical. And we need to pay them more attention, more care, more monitoring. And then those are the less critical cases. And lastly, those are, as we call it, they are in a stable condition. This would help. We can divert those resources or those limited resources to those patients who need it the most. Thank you.
Thank you very much. Thank you very much, Dr. Ola. So at the end of the first day here, I want to thank the organizers for their hard work. I want to thank our presenters for their lovely lectures and the presentations. They were all wonderful. And also, I want to thank our audience for their valuable time and attendance. And hope to see you all tomorrow at 5, Baghdad timing. Thank you.
For everybody.
Thank you, Doctor.
See you all tomorrow by 5 p.m. Baghdad time. Goodbye till then. Bye-bye.